Okay, so yeah, I'm just introducing Thomas Sarah from Brown University. So he's giving the next talk, and I'll hand it over to you now.
All right. Well, thank you, Sam. Good afternoon or good evening, everyone, depending on where you are. So this talk is going to be about trying to understand an interesting visual illusion known as the orientation tilt illusion. And so before I get into the weeds of my talk, because I don't think everyone here in this audience might necessarily know about the illusion, I wanted to briefly review it. So the perception of a center grading can be drastically altered by its surround. So on the left here would be a vertical grading, which should appear tilted to the left. On the right here is still a vertical grading, but that should appear tilted to the right. The illusion might not be obvious, because some of these effects are frequency and scale dependent. So depending on the size of the stimuli on your screen, it may or may not work. You might have to trust me on this. But here on the upper right corner are data from O'Toole and Van der Roth that have been replotted. So on the y-axis is the reported perceived shift in orientation from human participants. On the x-axis is the corresponding angular difference between the center and the surround. And so what you see here is that when the angular difference between the center and the surround is small, the surround appears to repulse the center, so tilting it away from the orientation of the surround. Past a critical angular difference, the effect reverses, and then the surround appears to attract the center. So a critical question for vision science is whether these effects or this phenomenon reflects a bug of the visual system or rather a feature. And so to give you a preview of some of the main conclusions from my talk, I'll try to make the point that this orientation tilt illusion turns out to be a consequence of recurrent neural mechanisms and circuits that are optimized for the detection of contours. In the process, I'll describe the work we've done in building a machine learning module, a recurrent neural network module that is grounded in the anatomy and the physiology of the visual cortex. And we will use that as to ask questions about the role of this recurrent circuits. And I'll show you that one of the major benefits of such a recurrent neural network module is that on visual tasks and on image segmentation tasks in particular, we're able to get a model that generalizes much better using much fewer training examples compared to state-of-the-art feed-forward neural networks. And then I'll conclude briefly by showing you that actually on one of the current kind of main challenge in computer vision, which is called the Microsoft Coco Panoptic Segmentation Challenge, we're able to actually outperform the state-of-the-art with this recurrent neural network approach. So just to give you some context for why our focus on recurrent neural networks, if you look at the state-of-the-art in computer vision, whether this is image classification, image segmentation, or action recognition, the progress has been steady for the past several years, but the state-of-the-art is still dominated today by feed-forward neural networks. I think part of the engine driving this progress is actually pretty clear. This is just a snapshot view of what has been happening on ImageNet. On the x-axis is the year that the challenge was being held. The height of the bars here correspond to the error rate for that year, and you see that the error rate has been decreasing steadily. At the same time, this red curve shows you that the number of layers in the feed-forward neural network that won the challenge on that year has been increasing, I would say, almost exponentially. So much of the power of modern deep neural networks is achieved through kind of static and what I would call brute force depth. In comparison, if we look at the anatomy and physiology of the visual system, the architecture is somewhat different. This is a view of the side of a monkey brain, the ventral stream of the visual cortex, which is involved in the processing of shape and object information. For almost every feed-forward connections in our visual system, we can find a reciprocal feedback connections. So if we look at the kind of underlying architecture and corresponding models of the ventral stream, in addition to the feed-forward connections that are well approximated and modeled in current feed-forward neural networks, our visual system contains local feedback connections. I'll refer to those connections as horizontal or lateral connections. They will typically run between retinotopically organized units within a layer of processing. And in addition to these horizontal or lateral connections, one can also find what I'll refer to later as top-down connections, so running from a higher stage of processing onto a lower stage of processing. Case in point, the depths of processing that our visual system is able to achieve is not through this kind of static sheer depth, as in feed-forward neural network, but rather the backbone of our visual system turns out to be a much shallower neural network, where the depth of processing is happening through time, through those recurrent kind of connections, and an interplay between feed-forward, horizontal, and top-down connections. So my lab has been trying to kind of bridge the gap between what we know about neuroscience and the role of recurrent connections and the reality of computer vision, which is still today dominated by feed-forward neural networks. And our starting point was a few years ago, where we tried to better characterize and understand the recurrent neural circuits that are responsible for the so-called extra-classical receptive field phenomena, which are very well documented electrophysiology phenomena. So maybe just to make it more concrete, I'm sure many of you in the audience are familiar with the notion of classical receptive field, which is the part of the visual field that needs to be stimulated for a particular neuron to elicit a response. The extra-classical receptive field is an area that typically sits right outside of the receptive field. If one stimulates the extra-classical receptive field by itself, it will not elicit any response from the neuron, and that's by definition. Otherwise, it would be part of the classical receptive field. But now, if we present something like a full field stimulus, or at least a stimulus that covers both the classical and the extra-classical receptive field, then it is known and well documented that the extra-classical receptive field can strongly modulate the neural response. It is known that, and widely assumed, that feedback connections, so both top-down and horizontal connections, play a role in these kinds of classical, extra-classical receptive field phenomena. We build what I would describe as a computational neuroscience model. It's very closely related to some of the dynamical system models we heard about this morning. Our goal was to build a model that would be able to essentially explain and model the interactions between neurons in the visual system, both within a cortical column. So think that maybe within this cortical column, we would have populations of neurons spanning a range of orientation preferences, or maybe a range of hue preferences, or disparity preferences, or motion preferences. So we're going to have to model the pattern of connectivity within that column, both excitatory and inhibitory, in addition to explaining how these neurons should wire together and connect together, knowing that our visual system is organized retinotopically, with those cortical columns, each containing the same basic dictionary of features, being replicated across position in space. So I'm not going to tell you very much about this model. This is published, and this is just really a motivation for the remaining of this talk. I'll just point out that there are kind of two key parameters in this model, which is this WI and WE, which characterizes the synaptic connectivity within the models. And so we essentially end up with a systems of ordinary differential equations, and when we present a full field stimulus, we end up and potentially model thousands of cortical columns. We end up with a model that includes, on the order of millions of differential equations that need to be solved. So that tends to be a challenge all in all by itself. But my former graduate student, David Melly, was able to essentially figure out how to tweak the pattern of inhibition and excitation, again, both within and across cortical column, using anatomical and electrophysiological constraints. So you look for all the monkey electrophysiology you could find on this extraclassical receptor field phenomena, and by tweaking the pattern of synaptic connections, he was able to get a model that would be consistent with the known electrophysiology. And then the main contribution of this work was now to test the underlying circuit with the kinds of contextual illusions that have been studied psychophysically. And what we found is that across visual modalities, we were able to essentially account for a pattern of psychophysics results and the perceived perception, the perceived estimates from human subjects during a variety of contextual illusions. So including both the orientation tilt effect that where I started from, in addition to color illusions, motion illusions, stereo illusions, etc., etc. Now, one of the issue, in my opinion, with this kinds of continuous time dynamics recurrent neural circuits is that I mentioned earlier, they are hard to simulate. And in practice, this means that it becomes tedious and very hard to actually optimize systematically the pattern of connections within these circuits. So we were kind of lucky in this first instantiation because we could use the physiology to constrain the pattern of synaptic connectivity. But in general, if we want to get at the level of understanding the potential role that these recurrent circuits play and whether those contextual illusions are bugs or features, we would like ideally to optimize this pattern of connectivity so that this circuit could solve almost arbitrary visual recognition tasks. And so we realized that we were using a very simple method, Euler integration method, to solve these differential equations. And we realized that essentially we were solving these differential equations using discrete time approximations, which could be completely learned and approximated with kind of modern recurrent neural network and deep learning tools. So shown here is an example of the actual unit we use, which is called gated recurrent unit. If you've never heard about gated recurrent units, that's okay. They are simplified units compared to the LSTM that you heard about this morning. The details are not critical. The take-home message here really is that we have the tools at the moment from machine learning and deep learning that allow us to essentially approximate these kinds of continuous time neural circuits. And the benefit of building mathematical or machine learning abstractions of those neural circuit models is that now we can essentially take those modules, plug them into our favorite deep neural circuits, and optimize them at scale to solve arbitrary visual tasks. So in order to demonstrate the benefit of these kinds of what I would call highly recurrent neural network modules, that is neural network architectures where neurons are allowed to communicate across neighbors within a layer of processing, as opposed to the feed-forward neural network that can only send information bottom-up. We came up with this task that we call a pathfinder. So I think the best analogy we could come up with would be a task where you would have to follow some footprints among many other footprints. And so the task was cast as a binary categorization task. On the top row here are examples of positive examples. On the bottom row are negative examples. Those images are black and white. You should see kind of contours made up of paddles. And then on both sets of images, there are markers that are these white circles. And again, I don't know whether you can see this at home. But the main difference is that on the positive set, the two markers fall on the same contour. So there is a path linking the two markers. There is no path, or the two markers are on different contours on the negative set. We never give the rule to the neural network, obviously. They learn, they have to discover the rule by learning from examples. Our hypothesis here was that our recurrent neural network would be solving the task by incrementally grouping neighbors, by passing information across neighbors. Feed-forward neural network are universal approximators, so we knew that in theory they should be able to solve the task perfectly well. if we have enough training examples. But our prediction was that the way they would solve this task would be by building receptive fields that encompass the entire contour. And so our prediction was that as we make the contours longer and longer, from say 6 petals to 9 petals to 14 petals, our expectation was that the minimal architecture, the minimal feed-forward neural network capable to solve the task should get deeper and deeper, because that's the way deep neural network grow their receptive fields. And so that's a prediction that we tested. So there is a lot of information on this figure, and that reflects the fact that our reviewers wanted a lot of controls and for us to test every possible architecture. But I think to some extent, I'm going to draw your attention to the top row here. What you see here in red, accuracy on the y-axis, and then we have the accuracy over different path lengths. Our recurrent neural network, which we called H-GRU, as in horizontal GRU, can learn the task at all contour lengths perfectly. So there is no effect of contour lengths on the performance of the network. I'll skip through a number of controls here. I'll bring your attention here to the ResNet, which is considered a state-of-the-art neural network. For small, short contours, we find that ResNet 1850 can solve the task. However, for the longest contour, 14, we had to go all the way to 152 layers of processing to solve what would seem to be an otherwise kind of trivial visual task. So if we replot this data, where here on the x-axis is the essentially the percent multiplier, so the number of parameters multipliers compared to our baseline architecture, which is the H-GRU. So here we have perfect accuracy with a multiplier of 1. You can get also very good accuracy with feed-forward neural network, but as I said earlier, we need to get all the way to 152 layers of processing, which leads to 1,000 times more free parameters to be fitted, which in practice means a network that will require many more training examples to learn as well. All right. What we've done since is to continue kind of extending and building up our recurrent neural circuits. We started with a model of the horizontal connections. We have since extended the module to allow for top-down connections. We have also devised tasks that would allow us to demonstrate a double differentiation between the role of horizontal and top-down connections, depending on the task. And I'm not going to tell you anything about it, but if there are people in the audience interested in the details, this was published again this year at iClear. What I'm going to tell you about today is the fact that now, again, we have a hierarchy of processing stages, and we're going to be training this recurrent neural network for the detection of contours on the Berkeley image segmentation datasets. So I should point out from the beginning that the level of accuracy of the state-of-the-art on this Berkeley dataset is already human level. And I should also point out that the state-of-the-art on this dataset are feed-forward, very deep feed-forward neural network. This is called BDCN here. And without dwelling too much on the details, I should say that the state-of-the-art is obtained and human level of accuracy is obtained through a lot of bells and whistle and a lot of messaging to be able to train those neural networks. So typically they are pre-trained on ImageNet for image categorization. They are further fine-tuned on a different data set that has only coarse contour information. And then they are further fine-tuned on this Berkeley segmentation data set. But because the data set is too small, both the training and the validation sets are used. So it's a lot of data and training messaging. Our recurrent neural network is able to also achieve human level of accuracy when we do all of this complicated kind of training procedure. But what is more interesting, I think, is what happens when we start cutting down on the number of training examples used by those feed-forward and recurrent neural architectures. What you can see here is that if we remove some of the data augmentation, which are some of the tricks used to get this very deep feed-forward network to train at all. So using the entire set of images, but no augmentation, we get a big drop from human accuracy. Our recurrent neural network, GammaNet, also get a drop, but it's much less so. And in particular, you see that here, our GammaNet is able to achieve the same level of accuracy as the very deep feed-forward neural network, that would have been using or being trained with 100% of the training data, where now the recurrent neural network was trained with only 1/10 of that, so 10% of the data. So the partial conclusion here is that one of the benefits of these recurrent neural circuits is to be able to achieve at least equivalent robustness and generalization while cutting down the sample complexity. So that is being much more sample efficient than their feed-forward counterparts. And to illustrate kind of the difference on how this recurrent neural network solved the task compared to feed-forward neural network, here is, so these are different images. At t=1, the network produces an initial set of predictions for the presence of contours. It's very noisy. There are many contours that are missing. There are many spurious ones. But through time, dynamically, through the exchange of information through recurrent connection, the network is able to fill in the missing information, so filling in missing contours, and then suppressing kind of spurious contours. And you see that at times t=8 here, we're able to get a final prediction from the network that is a fairly good approximation of the ground truth that is derived from human subjects. Now, going back to our original question about the role and whether the tilt illusion is a bug or a feature, I should point out that, so we run the same psychophysics experiment on this recurrent neural network optimized for contour detection. We find a qualitatively good fit with human data. I should point out that the effect is actually much stronger in the model than it is on humans. It's 1 order of magnitude larger. But interestingly, the feed-forward neural network does not exhibit any effect at all, despite the fact that those two architectures perform at the same level of accuracy. Actually, all three here would be human, GammaNet, and BDCN would be achieving the same level of accuracy on contour detection. So this leads us to believe that, one, recurrent mechanisms are necessary and sufficient to account for the tilt illusion. And two, the tilt illusion appears to arise as a byproduct of a circuit that is optimized for contour detection. And I'm going to skip this because I'm going to run out of time. I'll just say that we found a way to correct for the illusion. And we find that if we do this, two things happen. One, the accuracy of the recurrent neural network drop. So the contextual, the tilt illusion is really not a bug. It's something that is part of the inherent computation carried by the system, and that seems to be optimal for contour detection. I want to briefly give you an update on this work. Our current effort is in trying to continue scaling up our recurrent neural network architecture, and in particular to demonstrate their benefit on compared to feed-forward neural network on what are considered to be the very kind of large-scale computer vision challenges. So here's an example of such large-scale, computer vision challenge. This is called the Microsoft COCO Panoptic Segmentation Challenge. Here the idea is for the system to classify every pixel, to output a class label for every pixel. This would be semantic segmentation. Not only that, but when there are multiple objects, sometimes occluding each other or at least being in close proximity, the system cannot just have a single kind of class label for the entire group, but the individual objects, the different instances have to be segmented out. So this is a very hard task. The measure used to evaluate progress is this panoptic quality measure. The state of the art is in the lower 40%. And this is the current state of the art, a version of a ResNet, which I'm not going to tell you much about. I'll just say that compared to our recurrent neural network, it contains about 1 million extra parameters. All we did here was to add our recurrent neural network module on top of this feed-forward neural network. With just one iteration, one recurrent step, we can already get a substantial gain in accuracy. And then as we increase more and more steps, we start seeing improvements. Now, I should point out that one of the initial limitations we had with training those recurrent neural network at scale on very large computer vision data sets was the fact that the backpropagation through time, which is one of the main methods used to train this recurrent neural network does not do well with memory. That is, if we wanted a system that has a lot of time steps, we would need more and more GPU memory in order to train this network, which, given that we're starting from relatively deep networks already, this seriously limits how much kind of recurrent time steps we can actually compute. We came up with an extension of the backpropagation through time. Again, I'm not going to tell you the details. I'll just say that it works. When we use our extension, we find that now we can train the network with an arbitrary number of time steps, because we are not anymore bound by the GPU memory, and that we're able to get significantly improved results with 20 steps of processing. So just to give you a qualitative sense for the gap here between the feed-forward network and this recurrent neural network, I'll show you here just two examples. On the left are the feed-forward networks, right is our recurrent neural network. The colors of the pixels here correspond to the predictions of those models. You see that the feed-forward networks here hallucinates a sheep in the rocks. It hallucinates here a zebra in the wagon. And these are the kind of spurious detections that a recurrent neural network is able to suppress. I'll just show you qualitative results on this COCO segmentation data set. You see that the network behaves very differently from those feed-forward, hierarchical, very deep neural network, where processing would be done through static depth. Here, the network seems to learn, and this is not something we had coded in the network, it learned that as a strategy. I would call this strategy a filling-in strategy. The network starts with an initial seed on the object somewhere in the center, and then the information gets propagated until the entire object mask has been filled in. All right, I'm already at 25 minutes, so before I conclude, I just want to, I'm aware that the workshop really is as a health component to it, and I wanted to emphasize the fact that the approach we've taken to go from this neural circuit models, recurrent circuit models, to deep neural network modules that are trainable and optimizable from data can be broadly applied well outside of vision. And so, case in point, we are part of a large-scale DARPA project which aims at restoring restoring movement in patients that have had a spinal cord injury. We have colleagues who are developing electrodes that can be implanted above and below the injury site. Those electrodes can simultaneously record and stimulate directly the spinal cord. Our goal is to build a smart interface that would enable learning architecture to learn to reconnect the two sites. In general, you could do that with a black box, very deep feed-forward neural network, or black box, fully recurrent neural network. After all, those are universal approximators, and they can learn anything in the limit of a large amount of training data. Our approach has been different. Our approach has been that we think that if we approximate the circuits that we are trying to bypass here, will fare better. And so we have been able to apply this exact method, starting from a biophysical model of the spinal cord, essentially starting from a wiring diagram, and then building a machine learning approximation. We are finding that the underlying interface learns much more efficiently than a generic kind of black box type of interfaces. I'm way out of time, so I'm just going to leave this slide while I take questions. I want to thank Drew Lindsley and Rex Liu, who are fantastic postdoc in the lab, Jungkyun Kim, who is now at DeepMind and was a former grad student of mine, Lakshmi Govindarajan, current grad student, and Alec Ashok, who is a wonderful research assistant. Thank you very much.
Thanks very much. That was very interesting. So I have one question so far from the audience, and that is whether you could also model higher level feedback for attention or saccades from frontal eye field using modifications of the approach that you took.
This is exact. So we haven't done it yet, but this is this is exactly the thesis topic of one of my graduate students. So I completely agree. The next step is to incorporate attentional mechanisms and allowing, if not saccadic eye movements, at least being able to model shift of attention. Absolutely.
And is it possible to measure the kind of temporal dynamics you see in the biophysical kinds of models using your networks?
I'm sorry, so I'm not sure I fully understand. So to measure do you mind repeating?
The spiking dynamic is that there's no spiking, but the kind of
no. So this is So to be clear, and I probably was not clear enough. We're starting from rate-based models, so kind of there is no spike here. But I think in general, again, the approach shouldn't be limited to just rate-based model. And in fact, I think it would be interesting to try to expand towards spikes. I'll just share one of my provoking views to this crowd. I don't think spikes will be very useful for AI. I do think, however, that obviously they are important for characterizing mechanisms of the visuals of the brain and circuits of the brain. So it would be interesting to try to leverage some of those tools to build kind of machine learning approximations.
Thanks. Okay. Oh, there's one more question, just quickly. If you've looked for other visual illusions.
So we are in the process of, this. So we have, so the original model that was not trained or optimized, that was kind of tweaked with graduate student dissent. So this is a psych review paper from 2018. And there we have a end-tuned model, and that we found that it was able to explain a host of contextual illusions. So I've shown you the orientation tilt illusion. We have both color contrast and color assimilation. We have related illusions in the motion domain and in the disparity domain. Right now, we have only been able to optimize these circuits on contour detection. We are trying to extend. So I suspect that those illusions in color, motion, and disparity reflect or are the byproduct of recurrent circuits that are normally optimized for other kinds of tasks. So I think color induction is a byproduct of color constancy, for instance. And so I have a graduate student who is optimizing and training the network on kind of color constancy computer vision databases, and we should be able to report on that soon. And similarly with motion and stereo, it's a matter of figuring out what task to use to optimize the circuit in order to account for the phenomena.
All right, so our next talk will be Terry Sanyovsky, Salk Institute and UCSD, who will talk on strong inhibition and stable temporal dynamics in spiking networks, sustained working memory. Let's see, Thomas, you'll stop sharing, and then we should be able to get Terry on somehow.
Okay, I invited Terry now.
Oh, here we go. Share screen.
All right.
So I pressed share screen. Application window. Ah, okay. Here and share. And here we go. Can you see that?
Yes, I see it and I hear you.
Okay, you are looking out over the Pacific Ocean, and I really have enjoyed a lot of the talks here, and in fact, I couldn't have asked for a better talk to precede mine, because it's going to make a point that a major point that I want to make, and it has to do with how you go about modeling. It's a whole new approach. And the real focus here is going to be on working memory and developing models, network models of working memory, and specifically spiking models of working memory. Now, the bigger point here has to do with the use of these really powerful modern tools that have come out of machine learning, and Thomas really used them very, very powerfully to explore the visual system. And I've written a book about this called The Deep Learning Revolution. It's a trade book for a general audience. And the real punchline is the tagline at the top, which is artificial intelligence meets human intelligence. And there's a convergence going on now between these two fields as neuroscience has better tools and techniques for understanding neural circuits, and as the tools for analyzing data from machine learning get better and better. And we can now take these tools and understand better how brains function. Now, you can buy this book from Amazon for $22, or you can go to PNAS, just Google Sejnowski and PNAS. There is an article there on it's a kind of a Cliff's Notes of the book on the unreasonable effectiveness of deep learning and artificial intelligence. And it really was aimed at a scientific audience, and specifically the issues of what's underlying the power of mathematical reasoning, mathematical underpinning of deep learning is really the focus. And it has to do with the mathematical properties of high-dimensional spaces, which is counterintuitive. Okay, so if you go back in the literature, there are a lot of models for working memory, and one of my favorites is the recurrent network model. This is from a review paper, and Daniel Derstovitz, who gave a really nice talk earlier as one of the co-authors. He was a postdoc in the lab back in the 2000s. But it's a very simple model, and it's very appealing. The idea is you have a very highly recurrently connected groups of pyramidal cells, and if you analyze it, you can analyze it mathematically. You can also simulate spiking neurons, and what you see here on the right is
Terry, your slides are slightly off the screen. I don't know if there's anything you can do about that.
Oh, okay.
It's like they're too big, and they're off on the bottom and the right. Okay,
well, I am going to go off-piste and see if we can enlarge them here. Let's see. Let's go to full screen. Oh, that doesn't work. No, I don't want to close video. I don't know why that happened. For some reason, you are screen sharing. Click to close message. Okay. Let's try that again. First, let me go to here and enlarge it. Sorry about this. I should have done a little bit of testing earlier. I take it you're not seeing anything right now because I'm not sharing.
Just your face, yeah.
Oh, you see my face. Oh, okay.
Yeah, it's probably, you can probably go back to the way it was. If it was mostly there. I didn't know it would be this.
I think we can do a little better. I think if I can just tune this up a little bit. Now, can you see that?
Now I don't think you're sharing, at least at the moment.
I'm not sharing? Oh, okay. Then I must share again. Okay, how's that?
Okay, now I see. I see it as a little, there it comes. I mean, now we have the full screen, but it's in a non-presentation mode. It's okay. Either.
Yeah, I think we can handle.
That'll work.
Okay. Okay, so as this is a very well-studied recurrent network. It's really a Hopfield network, and it has two stable for a given input. It can have two stable states, one when a lot of units are active and one when they're not. And Xiaojing did some nice work showing that it's a stable you can stably store an activity if the synaptic time constants are long, namely NMDA receptors. So this is a very common way of going about a network, building a network model. You have an idea, maybe if you connected up the connectivity in a certain way and you fiddle with the parameters, you'll eventually get it to do something. And we've taken that a long way, but the question is, can we come up with new ideas that maybe we didn't have for connecting up the network? And that's why these learning algorithms have proven to be so, I think, important. So a lot of work was done over the last decade or two on training up recurrent neural networks. And we've heard about some of the exciting work that's being done with the recurrent networks, backpropagation through time. And so here's the traditional model where we have a activity, we have an input, so we have input connections, we have recurrent connections, and we have output connections. And you can train it to do an arbitrary input-output mapping. Now, what we're going to be focused on here is to try to transfer one of these recurrent networks that was trained up on a particular task to a spiking network. And here's how we did it. This was published in PNAS in 2019. And a lot of people have tried to make progress with this, and it turns out that there's a very, very simple way to do this, and it's a one-for-one substitution. You don't have to multiply the number of units to average out the noise from the spikes. What you do is you train up the recurrent network, as shown here, and this is a very simple task, which is simply to integrate the pulse coming in up to, as you can see here, up to the output. And if there's no pulse, no output. So it just detects an input by integrating the pulse. So you train that up, and now what you do is one for one, you have the network, but the connections have to be scaled, and the connection strengths of the recurrent connections and the outputs have to be scaled by lambda. Now, the performance of the integrate and fire network is shown below it with lambda equal 1, which is what everyone was using. And you can see that it fails miserably. It should be, the correct output should be 0 or 1. But if you scale it down by a factor of 50, it performs almost as well as a recurrent network, as you can see here. And here's some activity patterns. And so you can see it's quite feasible, and this works almost all the time, depending on the difficulty of the task, to be able to transfer. So we have a way of creating spiking networks now using the full power of training arbitrary rate-based neural networks. So now, okay, let's put in a delay task. This is a temporal XOR task, and the idea is we have two different inputs: a first stimulus separated in time by a second stimulus on the order of half a second. And if they're both the same, up or down, you want the output to be 1. But if they're different, you want the output to be -1. And now XOR is a really trivial problem. It was one of the first that was used for backpropagation. But what makes this difficult is that there's a time delay. And so somehow the network has to hold on to the information from the first stimulus in order to decide what to do for the second. Now, the other thing I have to tell you is that not only can you train with backprop the weights, you can also train intrinsic properties. And we, in fact, had each of the synaptic inputs train the delay. So here's a distribution of the learn parameters for the delay. And so you can go in now and look with principal components to see how the task was solved. And so on the first input, the green one here, you can see starting from the starting point here in the center, depending on whether it goes up or down, the trajectory of the internal dynamic goes to the left or the right. And then when the second one comes in, you get the bifurcation again and the decision. So this is great. So this is a good starting point. But now let's compare it to neural data. So the data set that we looked at was recordings from the prefrontal cortex. This is monkey data. Constantinidis has a repository where you can go and get a very large data set, something like 400 cortical prefrontal cortical neurons. And the delay match to sample task is relatively simple, but it requires a delay. And the idea is that you fixate, a cue comes on the left or the right, and you have to basically decide whether it's the same side or not by saccading. And so here are recordings from 150 neurons showing what's happening during the match versus the non-match. It's difficult to make any sense out of this, but the fact is that there's information here that we're going to extract. We reproduced exactly the same task now with our recurrent network, delayed match to sample. Here's the raster plot. And so again, just by looking at the rasters, you're not going to really understand what's going on in the network, but we can dissect out what's going on. With now the first analysis that we did both on the neural data and on the network data was to look at with the autocorrelation. And we did that by, first of all, binning 50 millisecond bins and then counting them for each trial. That's 50 trials, counting the number of spikes per bin. And basically with that bin size, it's only 0, 1, or 2. And then we autocorrelated the bins with different delays. And you can see that the time constant for the decay and averaging, of course, autocorrelation over the entire 1 second range here. But you can see that for these for spikes that it falls off after about 170 milliseconds with the exponential decay. So here's comparing now this analysis of the dorsolateral prefrontal cortex with the trained neural network. And you can see that they have a similar, in terms of the time constants, similar distribution compared to the untrained network, which has a very low time constant for autocorrelation. So the learning did create this internal state where it was able to hold on to the information over hundreds of milliseconds. And if you separate out those that are below the median with comparing it to the for the dorsolateral versus the recurrent, they're quite similar on the order of 70 milliseconds for the short and then about 130 milliseconds for the long. So we think we actually have a pretty good handle on the distribution. And now you can go even further. We can go into, because of the fact it's very hard to go into the prefrontal cortex and figure out what the circuit is, we have complete control and complete analysis. It's not a black box. We've created this recurrent network, and we have complete knowledge of the inputs, the activity patterns, the synapses. And so now we can go in and actually interrogate and find out what the circuit mechanism was. So first of all, let's look for, we need a measure, and we're going to use something called discriminability, and here's how it works. What we're going to do is look at the, and this has been used to analyze neural data as well. So we're going to be showing you both neural data and dorsolateral prefrontal cortex on the top and recurrent network on the bottom. So the idea is during, say, the cue and delay periods, can you predict, just by doing a regression from the activity pattern, can you predict what the first input is? Is it up or down? And the answer is that if you do that, there's a lot of information in the DLP, the dorsolateral prefrontal cortex of the units that we're using, and also during the delay period. So that's a really good indicator of that somehow in the activity pattern, it was preserving the input. And the same thing is true here, as you can see, where there's a lot of information here, both during the cue and delay period. Now, we wanted to compare this delay period with another network that didn't have any delay period. And so here's another task, alternative force choice. You give it a cue, and the output has to go up if it's up, or down if it's down. And it solved the task, and the time scale was much lower. You don't need long time scales. But now what we're going to do is with these, we have these two tasks. We notice that the inhibitory strength is a lot less. The average inhibitory strength is about 50% greater in the network trained to do the match to sample, delayed match to sample. So we went in and we scrambled the weights between the inhibitory neurons, and the time scale for the DMS goes way down. Whereas if you scramble the excitatory weights, it doesn't actually change them at all, the time scale at all. So clearly implicating the inhibitory neurons here somehow. And so, the second penny fell when we analyzed the inputs to the inhibitory neurons. It turns out that they fell into two groups. These are selectivity from the two input. There were some that responded well to the positive cues and others to the negative cues. So there's two subsets of inhibitory neurons that have both recurrent inhibitory inputs amongst themselves, each group, and also between. And you can see that the strength is greater between than within, and the number of connections is also larger within. By the way, some very important issue. We used the architectural constraint that we had 80% excitatory neurons in the model and 20% inhibitory. The more constraints you put in, the better the match is going to be to the data. And then when we varied the strengths of those inhibitory connections, we can see that increasing the strength of the connections between inhibitory groups increased the accuracy, and increasing the inhibitory strengths within a group decreased the accuracy significantly. So it's clear that there's some interaction occurring between these two groups. And so here is our model that we've extracted from the delay match to sample network. So the input comes in, and depending on whether it's up or down, one or the other of the inhibitory groups is going to turn on, and because there's mutual inhibition, it's going to turn off the other inhibitory group. Now, these inhibitory neurons are connected to a bunch of excitatory neurons, and if the inhibitory neuron pool is on, it turns off one pool of excitatory neurons, but this one is disinhibited. So you have output coming in through disinhibition on one set of excitatory units that will be maintained during the delay period, as you can see here. In fact, the firing rate goes up. So this suggests a completely different model for how you could implement working memory through pools of inhibitory neurons that are inhibiting each other. Interestingly, this very same model has been used for devising by hand small networks that can make decision making. And so there's an interesting convergence here. That is to say, there's a motif here that can do both things, can both do the decisions and remember things. Now, here's one last thing, not the last thing, but in terms of extracting out some interesting additional information about the biology. Here's a task where there's a context signal that tells you whether you should integrate the green input or the red input. And so you trained it up, and there's no problem doing that. But now the question is, if you start with a network that was trained for some other task, and you are only able to change the input weights, can you still do the task? Okay, so here are two networks that we trained up, one on the alternative force choice and the other one on the delayed match to sample. We took it and then we retrained, and we can see that it can do this. Both networks were able to retrain and do the task equally well. Okay. So let's do another transfer task. Let's try to train up on alternative force choice. And now we're going to see whether we can do a delayed match to sample. This is actually a non-match to sample. And we can see here that if we have the normal eye to eye, it basically is at 50%, which means it's a chance. You can strengthen it and get some performance up to about 60%. So what that suggests is that the strength of the inhibition actually is important. Now, if you start with the delayed match to sample, right, and so we have long correlation times, long the decay times, then there's no problem training it up on the delayed match to sample. We get up to 100% on most of the trials pretty quickly. So this suggests something. It suggests that if you start out maybe innately with these long correlation times in between the inhibitory neurons, now this mechanism, that you could basically handle a lot of tasks by just changing the input weights. And the question is whether there's any evidence for that. Well, first of all, we have data, it turns out, from the monkeys in the pre-trial period. What they did is before they trained the monkey, they gave it the same input, but the task was just to maintain fixation. So this is a way for getting out information about the strengths, the long time constants. And we can see here that the time constant for the monkey for this passive task had the same distribution as for the monkey after it was trained on a delayed matched sample. Now, going further, there's evidence in literature that the ratio of different inhibitory neurons varies from the posterior somatosensory cortex up here to the prefrontal cortex here on the left. And what you're seeing here is, first of all, parvalbumin density, as you can see, is very high. It increases going down. It's very high for primary visual and somatosensory areas, but is relatively low up in the prefrontal cortex. This is actually a mouse, so this is infralimbic, and this is the insular, agranular insular up here at the very top. And now what we can do is look at the somatostatin, the ratio of parvalbumin to somatostatin. So this is just a ratio now. We can see that over 80% of the inhibitory cells now are, the fraction of the inhibitory cells are somatostatin compared to parvalbumin. So it looks as if the somatostatin cells may be the ones that are doing this, subpopulations that are inhibiting each other, which is interesting. And that's a testable prediction. And I would have never guessed this if no one had ever told me this. Finally, and this is really exciting because this is another thing that can be tested, which is that if you look at the trial variability, it's different depending on where you are in the cortex and also in the recurrent network. So here's a case where there is low variability. On each of the trials, there's like 2 or 3, 1, 2, 3 spikes. But with high variability, you get some trials where there's like 6 spikes and others where there's none or 1. And if you do an autocorrelation, you can pick that up very easily. So here's the density, and we computed the Fano factor, which is a measure of variability. And you can see in the dorsolateral prefrontal cortex, there's a long tail here where the Fano factor is higher, and there's more short, small Fano factors suggesting that it's low variability, relatively low variability for the short ones. But here in the recurrent network, we have the same trend, namely for the long variability, this is the long time constants, I should say. The long time constants had higher variability than the shorter time constants. So we have the same trend. So it looks as if, in addition to this prediction, we predict that with the subset of interneurons that are responsible for the delay period activity will have high Fano factor. And indeed, this is what's seen in the dorsolateral prefrontal cortex data comparing to the recurrent network data. The longer the time scale, the higher the Fano factor. So all these pieces of evidence are pointing in the same direction. And so finally, you can do a lot of things. You can, for example, scramble the connections. And the only, this is now showing that if you increase the strength of the connectivity, the Fano factor increases in our recurrent network. Again, showing that it's the strength of the inhibition that seems to be controlling things between the subgroups. And here's, again, varying between -30 and 30 and showing this strong relationship. So this is another measure, Fano factor. So finally, let me just wrap this up and say that we have a strong hypothesis now. We have another model that might be contender for a very important function in especially in the prefrontal cortex, which we know is important for delay period activity and for short-term memory. We've been focusing here on the synapses and the neurons and the networks. I should say that this method can be used to train up not just the time constants, but also many intrinsic properties like membrane time constant, excitability, and so forth. So we can, by putting in more constraints and putting in more parameters, we can actually look for how each one of them are going to contribute to the performance. I also should let you know very briefly that we have another paper that's in review in Neuron, in which we can show that you don't even need the rate-coded network because we found a way to train up spiking networks and not only the synaptic strengths, but also the intrinsic properties. And with that, I will say that the student who did this work, Robert Kim, is an MD, PhD student, extraordinarily talented both in terms of doing these recurrent network models, but the other half of his thesis was on analyzing mismatch negativity data from schizophrenia patients. And so he's a double threat. And with that, I will say goodbye. Or take questions.
Thank you, Terry. Excellent talk, as always. I think we had a question from Sam. Did you want to ask something?
Yeah, that was very interesting to hear about the interneuron role to working memory. So I was curious what you thought about related work that was looking at prefrontal cortex slices where they found that blocking the HCN channel, the hyperpolarization activated channels, would block these kind of persistent graded firing in response to stimuli.
Okay, that is something we have not put in the model yet, but I will predict that, in fact, with the hyperpolarizing activated current with inhibition like that, it's definitely going to play a role. So that's something we can follow up. And the beauty of these tools is that any idea you have or any mechanism can be tested. And another meta point I want to make is, and this is Thomas's constraints, by putting in more constraints on the connectivity within a layer of the cortex, you can pull out even more and better performance. I mean, that's taking it one step further. You probably know that convolutional neural networks were inspired by what Hubel and Wiesel from that 60s era, 1960s, what they knew about the hierarchy and what they knew about the simple and complex cells. And so there's so much more we know now about the circuit properties and the interactions and the physiology in the cortex that this is really going to be going back and forth between the people who are studying brain circuits and people who are studying artificial circuits.
We have a question here about why variability is so high. Is it intrinsic noise or perhaps balanced network?
This is something that we haven't really explored yet, but in my mind, what's happening is you're looking at statistics of the arrival of clumps of inhibition that are going to turn off the unit for a long time, because some of the time constants are pretty long that were learned. So I think it's statistics, just statistics of the number of inhibitory inputs and whatever these spiking intervals are.
Do your results give any insight into potential applications to recurrent neural networks to reproduce brain motor cortex activity or other motor activities?
So I think that, so what's missing here? This is just a simple transformation from one temporal pattern to another. And that could be used as a building block for many, many other functions, including generating complex motor signals. We haven't done that yet, but I really think this is just the first. We're taking our first steps here, and it actually reminds me of what was like. Remember, Bill, back in the 80s when we had the first backprop results? We used simple feed-forward networks with one layer of hidden units to to try to understand population codes. And we used it in visual cortex to understand simple cells. This is Sidney Leckie, had a paper in Nature. And Anderson and Zipser had a really nice model showing transformation between retinal coordinates and head-based coordinates produced gain fields in the hidden layer. So in a sense, this is what we're happening today is basically an extension of the simple networks that we had already were giving us some insight, but they're so much better now, and computers are so much more powerful that we have almost unlimited. We're only limited by our own imagination now because we can really rely on these learning techniques to probe and to uncover and come up with new motifs, new network properties that maybe we were just maybe a lot more there. By the way, I really am completely convinced now that the inhibitory interneurons are really the major players in directing information within the cortex and organizing a lot of functions. And so I think we should have known that just from the fact there's so many different types of interneurons compared to the pyramidal cells, which seems to be innocent bystanders.
One more question was how your time constants were learned. Was that a backpropagation procedure to learn those?
Yes, it turns out that any differentiable function can be learned. That parameter can be learned with backprop. And anything that you want to throw in there. So but this is again, we just put in the ones that we thought were important, and they apparently were.
Great. Well, we can revisit some of these things in the discussion period later. Thank you. And Sam, you want to introduce the next speaker?
Sure. So the next speaker is Kenji Doya from Okinawa Institute of Science and Technology, and I'll invite him to the screen right now. Just a second.
Okay.
Hi, good morning. Can you see me and hear me?
Yeah.
Okay, great. So I'm very pleased to have this opportunity to join this workshop, even though it is 4:52 in the morning in Japan, and especially after Terry, who was my postdoc advisor in 1990s. Okay, so how can I share my screen? Okay, so this share screen button.
Yeah, so if you hover over your name, you should see the different icons pop up, one of which is share screen. Yeah, looks like it's working.
Do you see my PowerPoint now?
We saw it a second. Yeah, now we see it.
Okay, yeah. All right, so I'm from Okinawa Institute of Science and Technology from southern island of Japan. So the title today's talk is toward multiscale brain data assimilation. Okay, so yeah, so this is my version of how the brain theory meets big data. So we have some models about conceptual models of how the brain should be working. And these days, you have a lot of data about what the brain really is. Right? So we should eventually connect these. And then to do that, computational models are very important. Some of them are very abstract, functional level model, and some of them are very detailed biophysical models. And then you have top-down inference about how the model should work, and bottom-up data to constrain our models. And then in doing both direction of search, machine learning is very important, because the machine learning theories and architectures will allow us to come up with the candidates of the abstract functional models. And also the machine learning methods allow us to constrain our model based on the rich experimental data. So today I'd like to introduce some of the works my lab and collaborators have been doing. For example, several years ago, we did a project of application machine learning to psychiatry under the program called the Strategic Research Program for Brain Sciences in Japan. So my collaborators at the psychiatry department obtained lots of data from functional MRI and blood markers, including genotypes, and also many clinical data. And then we want to combine these variety of data using machine learning. So you can use a kind of unsupervised learning paradigm to identify possible subtypes of the disease, and also use a supervised learning paradigm to the diagnosis and also the prediction of the medication responses. So that was our idea. So there has been many studies going on in this program. So one recent report from our lab is to apply unsupervised learning paradigm called multi-view co-clustering to different data, including functional MRI and genetic and clinical data. And then we found that some of the subject cluster separated the depression patients and the controls, and within the depression subjects, some of them had drug resistance even after several weeks or months of SSRI treatment. Symptoms were not very good. So and then using this paradigm, we could separate the subjects first based on the functional connectivity of the resting MRI data around the angular cortex, and also their past history of like a childhood stress. So some of them can be predicted as a drug resistance. So this was a very early study with a relatively small number of subjects, but now our colleagues are increasing the number of patients to come up with a more reliable ways of subtyping of the subject and also diagnosis of the subjects. Okay. So another kind of more recent work we have been working on is in Japan's Brain Minds program. This is a flagship neuroscience program using marmoset monkeys. So this is the major main website, and we also have a dedicated data portal like a Brain Minds region JP. So here we have different kinds of data, like a basic brain atlas, and also we have some human data to eventually translate what we found in the marmoset brain to the human medical applications. And we have a high field, like 9.4 Tesla MRI for marmoset monkeys, and we performed the electrocorticography, so the brain wave was measured from the cortical surface, and also tracer injections and so on. And we also have the gene expression data as well. Okay. And So this project is going to get many different data about the structure of the brain and also the function or activities of the brain. But if those data are just stored separately, that is not very interesting. We have to combine these structural and functional data, right, as computational models. So, and the models can be at the different levels. Some of the like a macroscopic model, modeling the each population by the average fine rate, or spiking network model, modeling the spiking of individual neurons, or even subcellular structural models like conduction-based or signaling cascade. And ideally, these models with different levels should be constrained with each other. Right? And today, we are going to talk about some of our work trying to combine the diffusion MRI data with the neural tracer data, and also diffusion MRI data with the functional MRI data and the electrocardiogram data. Okay, so one project we performed is optimization of a fiber tracking from diffusion MRI data. Right? So diffusion MRI is a way of analyzing the brain in terms of the water diffusion direction. So if there's a myelinated nerve fibers, diffusion of water is a constraint to the direction of the fibers. So with that, from that information, we can estimate the local direction of the fiber running. And by concatenating those fiber direction, we can draw a kind of a global connection of the brain areas. But of course, the reliability of such a global connection depends on the tracking algorithm, and they have some several parameters. Right? So some of the parameters have a good basis from biophysics, but some of the parameters we have to optimize. And fortunately, in this brain management project, we have the both diffusion MRI data and also neural tracer injection data, which can be worked as a reference for optimizing fiber tracking algorithms. So we implemented a kind of multi-objective optimization framework to optimize these parameters of fiber tracking algorithms. And then by optimizing them, so we could see an improvement in the tracking, especially for the long-range fibers. And then the resulting connection matrix became different compared to the default parameter people usually use. And this is the actual implementation of our algorithm. So the Carlos, who did this work, came up with a first tested a single objective, like a true positive rate, but he proposed like a combination of 4 objectives, like a distance-weighted true positive rate, and the ratio between true positive and false positive rate, and also the region-wise connection coincidence, and also the passage of a long-range fiber right into the commissure area. And then by applying this multi-objective optimization algorithm, we could see these objectives improve over kind of generations, and also the average fiber length became longer by the optimization. And we optimized the parameters using 10 injection sites data, and then tested for 6 injection sites, which were not used for optimization. And then we could verify the good generalization of the performance, even for the data set for the brain regions, which were not used for optimization. So this can be used for the validation of the fiber tracking algorithms. And after making sure our fiber tracking algorithm is doing the right job, we try to use it to combine the functional MRI data and the diffusion MRI data. And from diffusion MRI data with the fiber tracking, we can come up with a connectivity matrix across different parts of the brain. So in this example, we parcellated the marmoset of the brain into 60 regions of interest, and then have a 60 by 60 connection matrix. And then plug such a connection matrix into a dynamic model. So in this case, we use a relatively simple rate model of a Wilson-Cohen type, so which assumes the excitatory population and the inhibitory population are connected with each other. And this excitatory population have a long-range connection based on the connective matrix coming from the diffusion MRI. And we have some parameters like a gain of this long-range connection and the strengths of interaction between the excitatory, inhibitory, and inhibitory to excitatory connections. And then by tuning these parameters, we can simulate the neural activity and compute the functional connectivity matrix. And from a functional MRI, we can also observe the Bordeaux signal and then obtain the empirical functional connectivity. And then we see further these two like features of the dynamics can match each other. And this is a framework proposed by Gustave Deco, first in human MR data. We are testing it with marmoset MR data with a validated connection matrix. So yeah, so this is the sorry, there's some error in the font, but we tested how the model behaved by the different strengths of excitatory connection and the inhibitory connection. Just as Terry told in previous talk, the balance between excitatory and inhibitory connection is very important. The model performance changes a lot by the balance of this excitatory and inhibitory connections. Right? So by setting some of the parameters, the functional connectivity produced by the model have a high correlation with the functional connectivity produced from the functional MRI measurements. So that is what we could show from this optimization. And we can also visualize the dynamic activities from the simulated model and observed Bordeaux signal. Okay, and we are further considering the way to combine the structural and functional data in the framework of data assimilation. So data assimilation is a kind of dynamic Bayesian framework which combines the dynamic model and multimodal, in general, noisy observation. And then this model gives a kind of predictive prior, and that is matched with the likelihood coming from the observed data to give a posterior distribution of the state of the simulation model given the available data. And that is used for the prediction of the model and also estimation of the unknown parameters. And this performance can be used for the improvement of the measurement methods. And this framework has been quite successfully used in weather forecast. People built a very large-scale dynamic model of the atmosphere using some kind of a partial differential equation, and combine them with meteorological observation, either from the satellite or the different equipment on land. and then make a best estimate of the current state of the atmosphere and use them for the prediction of the weather so several hours later or several days later. Right? And my colleague Ken Nakae proposed to use the same framework for the brain. So we have a structural model coming from a diffusion MRI and the tractography, which gives us a connection matrix. And we have observation of the activity from the functional MRI or electrocorticogram, electrodes placed on the surface of the brain. And then we can combine the model based on such connectivity data with the measurement by the MRI or E-Z in the framework of a data simulation. So when this model is linear, we can use a simple, like classic Kalman filtering. So when this model is nonlinear, we can use, for example, the particle filtering. So the method commonly used for like a weather forecast is called ensemble Kalman filtering, which is similar to particle filtering in terms of representing distribution by the collection of points. But update of the points are based on the Kalman filtering framework. So it has been shown to be scalable to high dimension dynamic model. So we have been testing this framework to this electrocorticogram with 64 electrodes placed on the cortical surface of the mammoth's brain. And then we can use a dynamic model based on the diffusion MRI. So either the high dimension model, like 1,500 nodes in the cortical surface, or more conventional regions of interest around 60. And the question is whether we can predict the high dimension brain activity based on the limited measurement of the electrodes. So Ken tested this framework by assuming a linear model of this high dimension cortical surface model. And he could show by the observation from the 64 points in the cortical brain. So he could simulate estimate the underlying activity in the cortical surface. So now we are trying to validate this result with functional MRI data. And also another colleague, Hiromichi, is working on kind of nonlinear data simulation using Wilson-Cowan model and the ensemble Kalman filtering. So this is still a very early phase, but for the 60 ROI model, we observe activity to only the excitatory neurons and use the data simulation to estimate the activity of the inhibitory neurons, which seems to be running fairly well. And even after we stop observation, we can continue prediction of the network dynamics for some time. And we can use this framework also for estimation of the parameters like excitatory inhibitor connection or inhibitor to excitatory connection. So this is a kind of very early but encouraging result. Okay, so let me conclude. So I showed some examples of data-driven approach, like human MRI data for psychiatry, and also the optimization and validation of the tracer fiber tracking algorithm using tracer and the MRI data, and also combining the structural and the functional data for understanding the underlying brain dynamics. So I think data simulation, which has been shown to scale well in other fields like meteorology, might be a very good framework for combining high dimension experimental data from the brain. So currently we are working on the only single level and extending it to multiscale is a very interesting future work. Okay, so thank you very much for your attention. And then this is based on the collaboration and the strategic research program for brain sciences and the brain minds program. Thank you very much.
Thank you, Kenji. So maybe we'll just segue directly into discussion and ask some questions for Kenji. Sam, meanwhile, you're going to invite the participants up, and if anyone else wants to be invited up, please just say something on the chat, and then we can do that. So what are the next steps, Kenji, with what you're doing here? How are you going to now apply this in a clinical domain?
Oh, yeah. So we previously applied the MRI data for the either subtyping or diagnosis, but that is only just mapping from the data to a class level. But we really want to understand the difference in the brain dynamics. So rather than just using classification, we want to characterize the difference of brain dynamics between the control subjects and patients of different disorders, not only the depression, but also schizophrenia or ASD and so on. So, and then having a deeper understanding the brain functioning in health and disease in a data-driven way is the direction we want to follow.
Has your data so far shed any light on the different subtypes of schizophrenia?
Not the subtypes of schizophrenia, but my colleagues in this program have identified the relationship between the brain dynamics of the ASD patients and the schizophrenic patients. So they have some commonality and the distinct part, right? So, but interpretation of the difference in the functional connectivity is still rather difficult. So one limitation is that we just use like a resting state MRI. So inclusion of the data during certain task MRI might give us a better insight about the brain function, not just during resting, but also doing some kind of the goal-directed behaviors. So yeah, there's still lots of things to do, but there are some kind of encouraging results coming up. Yeah.
That's very interesting. So let's open up the discussion. I see we do have Terry here, and he had a couple of questions from earlier that didn't get asked. Someone was asking about the biological experiments that you would propose to test what you've been measuring in your models. What do you do?
Yeah, very, very important issue. So one of the predictions is that the connectivity between the inhibitory units should have these subclusters or subunits that have strong inhibitory connections between them. And within the next, sometime later this year, we're going to have the connectome of a cubic millimeter of cortex. This is coming out of Clay Reed's lab. And so they'll have the connectivity pattern, and we've made the prediction for the inhibitory for the somatostatin inhibitory neurons in the prefrontal cortex. But there are a lot of other predictions that can be extracted depending on what the question is. Like, for example, we first noticed this phanofactor in the network. And fortunately, the data were already there, so we can test the prediction directly from the data. So those are the kinds of predictions that I had in mind. but these are very small networks, and in terms of the tasks that we're putting in, they're toy tasks, and so there's still a lot more needs to be done. In terms of scaling up, and putting in, for example, more choices, not just 2 choices.
And there was another question for you with regard to whether you can use multiple timescales during the delay period or just have a single timescale.
Yeah, it's up to you. You can pick whatever task or try. If you want to have a task that has 2 timescales, that could be learned as well. This back propagation algorithm gives you a lot of in terms of the timescales, multiple timescales. So that would be another step forward, is to include the time complexity as well as the spatial complexity.
So with regard to having further discussion here, it seems like we're somewhat limited if we're only allowed to have 4 people on at once, and we have like 10 people just on our panel, and then we have 100 and some people who would like to speak, no doubt. Has anybody used this app before? And have any idea how that can be done, or if it can be done?
You could put the voices of everybody up. The faces don't really matter.
I was thinking we could just do audio streams. At the moment, Sam, you can kick off at least Kenji and me, and you guys can invite somebody else up to talk. And we got various people here I know like to talk. Jim Bauer's here. Jim, you like to talk? You want to say something on our topic? I don't
go out.
Okay. So okay. Yeah.
All right, kick me off. You didn't even kick Terry off. Got anybody who wants to come in? Take them on.
So who's next for questions? Anyone? Jim Bauer says never.
Okay, can I ask a question?
Yeah.
So, Bill, you've been working on these very detailed compartmental models, and I was a little surprised that we didn't have a lot more of that in terms of representing the advances and how you've used those over the years. And so I wonder if you can expand on that, what your view is of where compartmental models are today.
Well, with respect to what we've been talking about here, I keep hoping for someone to tell me what I believe just instinctively to be true, that there are features that are critical about spiking. And I think you can speak to that from your simulations, that there are features that are critical about high amplitude oscillations with enormous coordination across cells, and that there are critical features about dendrites, which I think you partly touched upon when you talked about the difference between parvalbumin, which would be soma projecting, and somatostatin, which would be dendritic projecting interneurons. So I've continued over the years, decades, to come up with more questions than I have answers. So I'm happy to direct to you for answers, as I always have, Terry.
Well, I think that what's really missing from these deep learning models is the spatial interactions on dendrites. In other words, we know that branches can have a certain autonomy. We know that synapses are clustered and it can interact with each other. And so those are all computational mechanisms that are just crying for a computational role. And I think we can incorporate them in. I think all of these, there's no reason why compartmental models, they're all differentiable. We should be able to backprop right through them. So
we had hoped to have Idan Segev here to talk about his paper where he looked at a single cell and tried to develop it as a neural network. And Bartlett Mell has done some of that same kind of work.
Right. But they did it in the old-fashioned way, which is, come up with some idea for a function and then fiddle with the parameters until you get it to work. The beauty of our modern era is that, for example, for our delayed, delay period, when we got it to work, we expected that it would discover our smart, attractor model, because that everybody knows that that's very robust, and it was a lot of dozens of papers, including some of ours. And no, it found a completely different way of solving the problem. And I think as we put more constraints in with more mechanisms, that we'll discover other ways to solve problems that nobody had ever thought of before. So that's where I think I'm really hoping for more progress by combining these tools and techniques with what we already know.
Yeah, and in those terms, one thing that occurred to me as someone was asking about time constants, so they were asking a different context, is how you have these enormous numbers of different voltage-sensitive potassium channels, less enormous, but also a fair number of different voltage-sensitive calcium channels, voltage-sensitive sodium channels, and the time constants just spread out all over the place. And presumably there's some kind of a spectral advantage to having all of that's still underappreciated.
So, there's a really, I heard Rudolf Sepulcher give a talk on this. It was at Telluride, and he's a control theorist, and so he looks at neurons very differently from the way we do. We see them as a bag of ion channels, but he sees them in terms of feedback systems. And one of the really important points that he makes is that even within, say, neurons, that you have to, when you have a certain time scale, you have to have negative feedback in order to be able to control the positive currents. And the obvious one, there's a Hodgkin-Huxley axon, the sodium and delayed rectifier. But the same thing holds for the late after hyperpolarization and a lot of other pairs of currents. And so I think that some insight will come from understanding how each of those currents interacts and is there to be able to control homeostatically other currents. But he has a paper, and I highly recommend it. Just look up Sepulcher. Rudolf Sepulcher, very.
Sepulchre. I don't know.
Yeah, it's Sepulcher as in the tomb.
The tomb. Okay, that'll be easy to find.
Very unfortunate, but that's
poor guy.
That's a.
All right, we have Jim Bauer here, and I just want to make a historical note that there was a day 30 years ago when Jim and Terry would go head to head, and that was a lot of fun. So maybe we can get some excitement here.
Well, you should point out that it was Jim who actually founded this meeting.
That's right. And Jim has a historical book on this, so everyone should read that about the 20 years of CNS.
Yes, good to see you, Terry.
Good to see you, Jim.
Somehow we got old.
Funny thing, yes, that happens. If you wait long enough, it happens.
Yeah, but then what happens is you start pushing more and more data into a network that's more and more constricted in terms of its resources, and you end up coming up with solutions to the problem that have to do with understanding the nature of the problem rather than just lookup tables, right?
It's all in their tools, and you can use the tools well or poorly.
Exactly. So we had a whole conversation earlier today about why compartmental modeling seems to sort of be out of favor, or there are fewer examples of it at CNS this year than usual. And so I'm curious, Terry, why you think that might be true?
Well, I think fields go through ups and downs. And, just look at NIPS. Okay, so I'm the president of the foundation that's run that meeting for over 30 years. And, it was inspired by the first wave of neural networks, and Hopfield-Net was very inspirational back then. And we didn't actually, looking back, we didn't know what was going on, but what really was happening was, inspired by the brain, we went off into exploring the computational space, high dimensional space where you have a lot of interacting units, right? And big data. In other words, in order to get the learning to converge, you needed a lot of data. And we went as far as we could with the computing power we had back then, and so it kind of peaked. And then, but what happened was that a lot of other tools and techniques showed up, which were very powerful in machine learning to handle big data. And so, there was a period of about 10, 15, 20 years where, it went into exploding into many different directions, graphical models, support vector machines, which all, by the way, are used now in neuroscience, like for fMRI data. You heard from Kenji's talk, that this is, people who do fMRI they've used these tools now for decades. And so and now, but going back to the issue is that over the last decade now, the things have scaled up and computing power and data are all there. So now the ideas we had can now be enhanced. And for me, what's exciting is that as neuroscience goes into big data, and you can record now from tens of thousands of cells, 100,000 cells simultaneously, what are you going to do? You can't just do tuning curves the way we did it one cell at a time. And so we have to have ways of analyzing populations of cells. And it turns out that the tools that were developed by people in machine learning turn out to be good ones for being able to visualize and extract out population activity. So, so I think the same thing with the compartmental models. I think that, there was an early period where we were just trying to understand dendrites in some fundamental way. And over the decades, we've actually made a lot of progress, and we understand a lot about dendrites. It's still a lot to do, but I think the second era, which is now just beginning, which is to try to use computational tools to go in and query what's happening. What is a dendrite computing? What's the complexity of the information that can be decoded by dendrites? And so I think compartmental models have, they're going to come back. I think that is just a matter of again, these maybe the generational, maybe it has to do with a new group of people coming in with the same excitement we had back in the 80s. So at the NeurIPS meeting this last December, we had 14,000 people show up. I don't know, how does anybody know how many people have shown up here at this CNS meeting?
I think about 2,000 registered for the meeting.
Okay.
So, which is a good thing. Yeah, as you know, I was one of the first organizers of the NIPS meeting way back when.
Yes, I went through some of those old papers recently.
Yeah, so I think Yasser Abu Mustafa and I organized it for the first couple of years, way back when.
Right.
Anyway, so yes, was interesting time. My question about that is this. So we all agree sort of fundamentally, I assume, that neurons represent sort of the computational base for how brains work. Do we accept that? Sort of some version of the neuron doctrine.
Well, I believe in multiple levels. I think that single neurons definitely capture some fundamental unit, but I think that.
By the way, I'm not saying that single neurons, that by listening to a single. I mean, I was also one of the people that invented multi-unit recording back in the early 80s, right? So I deeply believe in that for a long time, that you can't understand a neuron outside of the context in which it's in, and that context is both the network plus the other neurons are involved. So, deeply believe that. But the question is this, sort of, if you're going to discover function, which is quite a different thing than, say, making a model that explains to people what you think the function is. If you're going to discover function, in the nervous system, what level of modeling should you do to do that? So, and just as an aside, and some of you heard me talk about this before, there are many models at many different levels that can replicate data. Probably the most successful model in the history of the world for replicating data was the model of Ptolemy, which explained for 200 years after the introduction of Copernicus, more accurately than the Copernican model, where you could find planets in the night sky. So replicating data is a good thing for models to do, but it isn't really proof of anything. And maybe one can argue that the further you get away from the physics, or the further you get away from the sort of computational or structural base, the more likely you are to generate models that replicate data but don't tell you anything. So there's pretty good evidence that the inverse square law came out of the fact that Newton actually made a model of the moon going around the Earth, and then invented or stole the calculus to actually, and found out that the force he measured was roughly, and it wasn't exactly right, but roughly inverse square to the distance. So that was a case where he built a model that was based on close to the physics, and out of it discovered something. So this was a conversation we had earlier, so I'm sort of summarizing it. My concern about the move off in the direction of more abstract models, and this has been my concern forever, and Terry and I have talked about this forever too. Okay. But my concern about moving off into the area in the direction of sort of more abstract models, bringing in machine learning, you can replicate data. The question is what you discover. And my guess from experience is that if you want to discover, if you want to discover how the brain computes, you somehow have to be really connected to the elements that do that. Okay, that's not to say that the networks don't contribute, all of that, but the element that ultimately sort of is making decisions based on what's coming into it, as far as what goes out of it in a simple form, is a neuron. And if you are and I'm making a statement of belief, if you're hoping to actually let the nervous system itself tell you what's happening, which is not a lot of what computational neuroscience kind of does these days, I don't think, but anyway, then you need modeling at the level that has the physics, enough physics in it, to be able for that model to say, aha, look at this. Okay, it turns out the parallel fibers don't do what people think they've been doing for 100 years.
Right. No,
that's what you need.
I completely agree with you. There's no doubt that there's a lot we don't yet know, and that let me give you one concrete example of something that I think was an important advance that hasn't really been absorbed yet by the community. So about a year ago, there was a paper that came out in Science. It was from Carandini's lab and Harris and Carandini. And what they had recorded from was from, I think it was like 40,000 neurons simultaneously using neuropixels in the visual cortex. And the first experiment was simply to look at resting activity, which in fMRI has become a big business too. And so they recorded all the spontaneous activity going on in a mouse that was head fixed. And they also did the same experiment with optical recording, got the same results. But what was astonishing is that at the same time that they were doing the recording, they were videotaping the snout of the mouse, the face. And it turns out that the mouse is always moving its snout, it's breathing, the whiskers are going back and forth, and so there's a lot of motion. And so they analyzed, using optical flow, which parts of the face were moving in what direction, And then they did a regression. I think it was a simple linear regression. They asked, the spontaneous activity in the cortex, could we predict the optical flow of the face? And the answer was yes. It could explain half the variance. That's a motor signal. And vice versa. If you take the optical flow, you can predict the spontaneous activity. Okay, what that's telling us, the spontaneous activity is not just noise.
Right. Well, Terry,
Are
you should
you-
be muted, or maybe I just can't hear you.
No, he was muted.
Oh, I'm sorry.
Yeah. So, yeah, but and this is a conversation we've had before, online and otherwise. I think the word noise is vastly overused and wrongly used in neuroscience.
Right.
Noise is how you explain things that you don't understand.
Completely.
But the core, one of the core insights of Shannon was that if you want an optimal communication channel, okay, in information theory, between 2 points, you want, if you're building the coding for that channel, you want the coding, the end result of the coding, to look as much like the full spectrum noise on that channel as you can. That is optimal communication. If you're using a channel, if you put full spectrum noise in one end, you look at what comes out the other end, and then you use that as the way to design your coding. So if you look, as you know, Ed Posner, sorry, we're old guys. Ed Posner, who was one of the core guys involved also in starting the NIPS meeting, as well as actually the meeting in Santa Barbara, which I think is the first time you and I ever met, was the meeting in Santa Barbara, right? Anyway, Ed Posner designed the communication system for the Voyager. Okay, and if you look at the signals coming down from the Voyager, if you do not know the coding, it looks like noise because it's efficiently coded. So in the nervous system, we record from neurons, which many have this beautiful distribution that looks at all the world like a Poisson, distribution. And then we declare, aha, there's a lot of noise, and we got to deal with noise, and blah, blah, blah. The ultimately, and I've said this for years, we will get closer to understanding neural coding when we peel away the noise excuse and start actually understanding that the tremendous amounts of apparent noise probably just represents the sophistication of the coding in the brain. And let me just toss out one other thing while we're at it. As you know, I've studied the olfactory system for a long time, and as you probably know, and you knew this was going to come up, I'm sorry. I see you smiling. Anyway, I actually personally believe you should never go to visual cortex unless you spend a lot of time working in olfaction.
And that's a whole other conversation, Jim. So
it is, but let me just say,
maybe
because
let
it's
the
important
old guys
hang on.
a chance to say something. Gunnar here, look how young he is. He is bald, but he's young. But he's unwrinkled. He's unwrinkled.
Yeah.
You're bald. And you're
Gunnar,
not
what did you want to ask?
Yeah, well, I have a couple of things. So first of all, this is basically the first time I'm interacting with this community. I'm very happy to be here. I come more from a general modeling of biological systems, where we have constructed rather comprehensive models for different organs, from sort of intracellular level to whole body level, how they are communicating. And as I presented before, we have done some models for some aspects of the brain. But I think I could come with some inputs on how we have managed to do the maybe comparatively complex task of modeling the body as a whole, but not specifically the brain in all its aspects. And one of the key things that we have really used in subdividing the modeling problem is to identify subsystems where you can measure the inputs and the outputs. So for instance, for organs, you can measure the blood flow that goes into the organ and the blood flow that leaves the organ. And then by knowing sort of what the organ is exposed to, and then seeing what it's doing to the blood, so you can measure the concentrations and so on, then you can consider the modeling of that organ as an isolated problem, which means that you can sort of go ahead and model that for as many years as you want and come up with first simple models and then increasingly more complex models, all the while knowing that all of these different submodels will fit together with the models of the other organs because the input-output profile is correct. And in that way, we have sort of been able to subdivide the enormous task of modeling the body as a whole in different groups that are focusing on different organs. And we can put the models together in the end because we sort of have the proper subdivision of tasks in a way. And one of the things I would like to know is, have you thought about similar ways of subdividing the task of modeling the brain?
In terms of metabolic requirements, is that what you're pointing out?
No, he's asking in terms of function. And the problem is that the kidney, you know what it does. Okay? And you can define what the kidney or what the heart does using well-described sort of either from chemistry or from engineering ways to characterize that. We don't know what the brain does at that level. Not close.
Well, I mean, there are different aspects.
If you look within I know, but if you look within the brain, for example, we like to assign tasks, like the cerebellum is a motor control structure, which it probably isn't, but we have assigned it that task for 150 years. Okay, but exactly what does that mean at the level of the modeling you're doing? What is the input and what is the output to be able to do the kind of modeling that you're talking about? We do not know. And it's a very hard problem. And I would, by the way, I'd just add one other quick thing. If you're building models of the brain or of the body and you're not including what the nervous system does in its control, eventually you're going to have to work with us.
Yeah, of course, and that's, I mean, one major reason why we have the brain, and what we have been doing primarily is just in neurovascular coupling, which is sort of takes electrical activity as input and then calculates how that is propagating through the different signals or cells that are involved in secreting vasoactive substances to control the blood flow. But what we haven't done yet, and this is basically what I'm hoping to get out of this meeting, is to find connections with people who have sort of the beginning of that process, the electrophysiology, and because we have lots of predictions of what the different cell types must do in terms of electrophysiology to create the correct time profiles of the vasoactive substances. And I mean, for me, it would be very interesting to hold that up to corresponding models where you model the electrophysiology much more in detail. And I think that there are similar physiological functions of the brain, which are very much similar to modeling other organs, while there also are these more sort of esoteric and magical things like learning and so on that we sort of don't really know if we even have the right concepts or
Jim, my apologies. I'm going to kick you off to let someone else on. Sorry.
I'm used to it.
Yeah, me too.
So Gunnar, I have something to add, which is that, for many years, I would say that we're studying the brain, which is the most complex device in the known universe. So my wife, who is a MD PhD, took me aside and said, well, Terry, you're wrong. She said, the body is the most complex device in the universe, of which the brain is a subset. And I think that, what Jim's point was right on, which is that the brain is monitoring everything. In fact, the insula has a complete representation of the viscera and is sending signals back and forth. And it's a very, very complex dance that is done, which I think is actually in some cases may dominate the simple control models that we build of the body.
Yeah, that's
but
true.
in any case, I think what you've done is magnificent in terms of setting up a system that allows you to put together pieces and be able to make predictions, and then ultimately understand how the complexity of the body is actually organized.
Yeah, and I think that there are things that you could do if you sort of think of it in that way. I mean, but the problem is always the data. I mean, is there somehow an ability to create a subsystem where you can measure all the inputs and then see what the system is doing? If you have that, then you sort of have a well-defined subsystem that can be considered as a
in some ways, it has been possible to do that, say, in the visual system.
Mm-hmm.
You can record from single neurons and now arrays in the retina and see how the information is encoded in the ganglion cells, and then how it's transformed in the lateral nucleus in the thalamus, and then it goes to V1 where it's transformed yet again, and then it's transformed up to V2. There are feedback connections that Thomas showed, which are incredibly important, haven't been as well studied. But yes, we have input-output models of each one of those cortical areas in the visual cortex. So it is possible. The farther you get in, the more complex it becomes in terms of understanding the signal. And there's a lot of information that has to do with the intracellular signals that aren't as well studied, dendrites and so forth. So there's but yeah, we're trying to do that, and that's really what neuroscientists have been trying to do for decades and decades and decades. It's just more difficult because the things are ultra miniaturized and they're very heterogeneous. There are many hundreds of different types of cells. The connectivity is very we don't even know what the degree of connectivity between all the different subtypes. So yeah, we would love to have that level of understanding.
Daniela, you want to say some things?
Yeah, I wanted to profit from the opportunity to have people with such a long experience in the field. So Terence, we talked at December last year, maybe you remember, in Vancouver. And so one thing that is haunting me in my comparably very short neuroscience kind of trajectory is, what if the cognitive notions, the notions of functions that we just happen to have, which have a long history in psychology and so forth, what if our semantic notions of the tasks that we use are incongruent with what the brain is computing? So, whether we study the whole brain with like MRI type of techniques or single neurons or groups of neurons, I think it's quite concerning to think that affect, attention, these types of things, they may just not cleanly map onto the brain. So that's also the topic of the new book by Bujaki. And that's this kind of the second part of my question. If that is not the most kind of solid aspect about the brain that we can take for granted and build on, what are the aspects of knowledge that we have about the mammalian brain that are most kind of solid and robust to build on?
Okay, so first of all, I completely agree that what we have right now is an embarrassment of riches, in the sense that we can collect data at a faster rate than anybody, terabytes per experiment, petabytes. And now we the rate limiting step is how you analyze it and the questions you ask. And unfortunately, we're saddled with a nomenclature that goes back to the to early 20th century. The words you use, attention. What does that mean? It turns out that if you look into the psychology literature, it turns out that everybody who sets up an experiment is using that word in a different way. There is no attention. There's this multiple mechanisms. It's a dynamical system that behaves in certain ways, and those words that we use are very slippery and have held us back. And so
but that sounds like a latent factor problem then. So let's say if you throw in a bunch of candidate tasks, let's say we can agree with experts on a battery of 20 tasks that we think are closest to what the brain does, maybe, and we say, even if they're built on semantic notions, psychological principles, concepts that we do not think congruently map on the brain, then at least they will probe these kind of unknown entities of processing that we don't know from different angles. So that would lead to some form of a latent factor approach.
So,
but that doesn't resolve the question, because the latent factors will still be unnamed.
That's a good starting point. And by the way, it is being done. It's a very nice brain-machine interfaces have now been developed to a point where they can be used to control robot arms. And Dave Susilla did a really wonderful analysis using autoencoders and looking at the patterns of activity in the motor cortex through an autoencoder and extracting out the latent variables in the middle. And he was able then to map those onto the arm motions in a much cleaner way than just doing a linear regression. So I think that is going to be very revealing, and interpreting those latent variables is where we will get beyond the simple names and semantics that we've had to work with for so long. I think that you put your finger on something which is missing right now, which is that ultimately we will have the brain is a dynamical system, very high dimensional space. And I've said this at the beginning of my talk. The properties of dynamics, of in fact statistics, of high dimensional spaces is something that is counterintuitive. In fact, deep learning should not work. Statisticians, optimal control theorists, they told us that you're crazy. You have a model here with thousands of parameters, and you're going to get you're going to be overfitting. You're going to get caught in local minima, and we're very naive. We went ahead and we didn't. We managed to have enough data, and we didn't get caught in local minima because it turns out in high dimensional spaces there aren't any, except when you get to the very bottom, when you're doing gradient descent. And so what's happening now is that statisticians are jumping in, mathematicians, and they're discovering properties of these high dimensional spaces, which are really totally unexpected. And I think that's the mathematics that will eventually lead us to the insights about how very high level representations in the cortex are used that have the ability to form concepts, language, and thought, and it's it's going to come through mathematics, through lots of recording and analysis of data using and these latent factors that you're talking about are going to be the gateway. That's going to give us an experimental access to the brain states that are important for behavior.
But still, what are the things that are most robust, that we can rely best on, that we already know about the brain to make forward in the direction you suggest?
Let's see. I'm going to pick up something else. I think that you actually had a very important insight. Almost 99% of all the experiments, the only exception I know are people who study the hippocampus. But what they do is they strap a monkey or a rat down, and then they train it up on one task to do something, to press a button if they see something. And the problem is that it's very under-constrained because the same neuron could be used for many, many other tasks. And so, if you see that it responds to a visual stimulus, you say, this is a visual neuron representing that stimulus. And the fact is that if you have many tasks, you'll discover that the very same neuron is involved in many different tasks. And it's high dimensional. Behavior is high dimensional. And neurons have inputs that are very high dimensional. And so understanding how that is how those maps come together and how the different how this very same set of neurons is participating in many different tasks. That's where we'll understand, the things that we hope will help us get to the bottom of how the brain is responsible for behavior. It's a it's a high dimensional problem, much higher than anybody imagined.
Daniel, do you want to say something more about noise, qua noise?
Oh, I think one man's noise is another man's signal, and I completely agree with Jim there.
All right, but now Daniel's going to say something.
Okay.
Yeah. So I throw in my 2 cents on noise here. So I think that noise is essential for cognition and for computation. Noise is a difficult term, of course, I acknowledge that, and its relation to information. But I think that, first of all, I mean, our world is full of uncertainty, and animals have to deal with that uncertainty. They have to compute and update probability distributions all the time. They have to do inference on them. If they want to make decision making, then in deep learning we have stochastic gradient descent. We inject noise on purpose for good purposes, right? Because we want to get out of suboptimal solutions, for instance. I think that animals are doing that all the time for decision making. I think it's a common observation that even if animals know what the rewarding option is in a simple two-lever task, lever task, that is with just conditioned operant conditioning paradigm, they systematically, they randomly probe the other option all the time. So they do inject noise on purpose, I think, all the time. And and I also think that biology doesn't try to avoid that. I think that probabilistic synaptic release in the cortex is one example. We know that synapses can do much better, but they do function probabilistically in neocortex. So I do think there's a big role for real stochasticity in the system, both at the computational, at the cognitive, and at the biological level.
So your point about the probabilistic release is very much similar to dropout in deep learning. As a way of regularizing. In other words, if you have too many parameters, if you turn off, originally it was turning off the unit, but now you can connect drop too. You get better generalization. So that's a good example where noise and you reduce the energy load, right? You don't have to have a release every time because that's expensive. But by the way, your example of lever pressing, actually we know a little bit about that, and because if your goal is to survive in the world, you better not make the assumption that it's stationary. That is to say, just because something worked today doesn't mean it's going to work tomorrow.
Exactly. Yeah.
So in fact, you do want to do some exploration, just to test to see if anything has changed, because that might be important for changing your strategy or trying to optimize your future rewards. And that's something that also comes out of temporal difference learning, which is a very popular model right now for dopamine neurons in the basal ganglia.
Right, but in a deterministic certain world, you wouldn't need that, right? So you need it because conditions are changing all the time, because you do not know. You have to work with probability distributions all the time in cognition, right? Right,
and that is the insight: is the world is probabilistic, and if you don't adapt to that distribution and that's changing, you're toast. And by the way, the evolution is all about doing that too. Is that what you need is if every animal had the same genome and you were monogenetic, if the environment changes a little bit, your species is gone. But by creating diversity, so that if the conditions change, there will be some subset of the genome that is able to adapt to that new niche, and then they will be able to propagate.
Yeah,
that is an essential part of all of biology is having probabilistic distributions and diversity.
Okay, I'm inviting Piotr Franachuk to say something. Again, this is a little unwieldy that I have to kick people out without even telling them, and then invite another person on because I'm only allowed to have 4 people at one time.
Actually, I'm close to bedtime, so I'm happy to drop out again.
Piotr, I invited you on. You have to accept, I think. I'll try again. Okay, we got Piotr.
Can you hear me?
Yes.
Yeah, because I had some problems with the microphone. No, I think this is a great discussion, and especially when we were talking about noise. I just heard on this meeting somebody talked about it, that infants actually can hear sounds on the level almost of the thermal noise. And the point is exactly that. I think our brain and the system during the both evolution and development in the individual is exactly picking up how far from this noise, which is not carrying any information, we can get, and then how close we should be to this level of noise to actually save on energy and all of the expenses. And the other thing is obviously that if we are talking about from the analysis point of view, about some other methods are talking about this is a background noise. Okay, this is the something like this. This is basically semantics. We are talking about that signal which we are interested in. Okay, so we are interested in the signal, the rest might be background noise. It doesn't mean that this is noise. I actually was working, and some of you know, on using stochastic models, autoregressive models for analysis of EEG. In this case, obviously, the traditional stochastic approach is you are saying that you are describing the system as the stochastic autoregressive model, and then actually everything rest should be white noise. Obviously, in EEG, it's never white noise. It's exactly that you can describe the part. I view it like this. Okay, we're interested in this part, which is stationary stochastic. We can use it later even for Granger causality or whatever. But then what we leave out is obviously not a white noise. It has the information. And this information should be analyzed by different methods. So that's the point, that actually it's misleading.
Well, it's wonderful that you talk about EEG, because that's something that we worked on quite extensively in the 90s. In fact, it was independent component analysis that was developed by Tony Bell in my lab, a postdoc. Yeah. That has been extensively used, as you probably know, for analyzing EEG independent components.
Right.
But one of the interesting things about ICA is that it only works if the signals are not Gaussian.
Right, right.
In other words, there's no way you can disentangle mixed signals if they're all Gaussian. You need to have something that is, and in fact, almost all the signals that carry information are Laplacian, that have long tails.
Uh-huh.
And that's perfect, because that signal can be separated out beautifully with ICA. And so it turns out to be a generalization using higher order information that you don't get from just the correlation, the second order moments. So that's a beautiful example of how you can go beyond the sort of traditional spectral analysis, which again is based on correlation. Spectral analysis is not giving you higher order information, and it's all, the brain is all in the business of higher order analysis. So here's another analogy I like to use. This is actually in the context of understanding how single neurons work by recording one at a time. Okay, suppose that you had the task of looking at the world through a soda straw, one pixel at a time, right? Mm-hmm. How much progress do you think you're going to make? Figuring out what's out there, right? The complexity of objects and all the things that we automatically are able to parse out from the world. But it's even worse, because the world is constantly moving and changing, which means that if you're looking at one pixel, it's constantly changing. So what are you going to get out of that? And so the information is in the population. That's where you got to find it, and it's high order information, which means that you just can't use the traditional tools that have been used. And by the way, I was really impressed. I've forgotten who gave the talk. It was Danilo Stock, DOZ. In any case, I think what he outlined there is something that happens all the time, which is that certain fields get stuck in a certain way to analyze the data. P less than 0.05. Yeah. I mean, that basically in psychology, you can't get a paper published unless you do that significance analysis. And it does have its use, but the point that he was making, which is that it's not going to help us to try to make sense of individuals. There you're making a prediction, for an individual, and that's a different question from significance. But
Munar, did you have some more questions asked?
I really like this discussion, and there is somehow almost a parallel discussion going on in the chat, which is a bit amusing to see. There is somehow crosstalk between them, but not always. And in the chat, talking about different types of models and models versus metaphors and so on. And this drives a little bit to my question, which is many of these things that we're discussing now are sort of quite fundamental. That they are sort of what approach should we use at all, and do we even have the right concepts or the right tools to understand what learning is and what are the processes involved and so on. I want to put that a little bit in, again, for me as an outsider, the sort of impression you get as an outsider from these enormous networks that exist. There's one in the EU and there's one in the US, both with $1 billion each, which you somehow as an outsider would think that there would come something out of it, which is not just sort of individual papers, but that is somehow a bigger picture of the brain as a whole. And that on the sort of level of understanding we have now, with the tools that we have now, with the concepts we have now, to produce something like that. In my field, there are corresponding networks on the liver and so on, which have had similar ambitions and succeeded to some extent. And my question here is there a strategy at place for producing something like that, something that is clearly beyond what individual researchers or collaborations can do, but that really requires this $1 billion?
Right. So I can speak for the BRAIN Initiative, which is the American BRAIN Project, because I was on the NIH advisory panel, which put together the BRAIN 2025, and that was specifically our goal. Our goal was to try to transform the neuroscience community in two different ways. One, by bringing in engineers who can build probes and tools and techniques for analysis, data analysis, that went way beyond what could be done with the technology that was being used. As I said, recording from one neuron at a time was basically the standard. If you go back to the 60s, right, that was a big advance. But the soda straw analogy means that you really need much larger populations. You need much more complex behavioral tests. And okay, so where are we? Okay, no, the second thing, which is what you're bringing up, which is that the tradition in neuroscience has been the R01, which is the single lab. And they do have program projects, which is supposed to be for multiple labs, but the vast majority of all of neuroscience is done with a single lab model, where every boat is its own bottom, which means that you have to be able to do all the tools and techniques. You have to be able to do all the analysis in your own lab. And for when you're exploring, that's a really great way to do it, because you can explore in many different directions. Many different labs can look at the problem differently, and that's great. But once you have a sufficiently powerful technology, then no single lab can actually use it anymore. So you necessarily need to have a consortia. You need to have ways of being able to coordinate, and that is what's happened. Actually, I was there's an annual BRAIN Initiative meeting, and I have to say that it has delivered something that I thought would never happen, which is that first of all, the engineers have been embraced, and the tools that are coming out have just exploded what we can do. Single cell genomic analysis, single neurons. We have, in fact, I was on a methylation project that looked at the methylation pattern of different neurons that distinguishes between different cell types. And the other thing is that there are the large consortia now that have formed, which and a good example of that, the early model was the Allen Institute for Brain Science. So this was funded originally, I think, for $100 million by Allen, from Microsoft fortune. And what they did there was very interesting. Was they started out with anatomy. They said, we've got to scale up, instead of each lab looking at one particular part of the brain and saying what the neurons look like or how the tract is between here. Let's do it on a larger scale. We're going to do all the tracts. We're going to do all the neurons. But in order to do that, we have to scale up, which means that we have to optimize the tools and techniques. And so they basically, took things that had been put together by hand and used differently in different labs and weren't reproducible, and basically optimized it to the point and automated it so that they can get very reproducible results and scale it up so they can do a whole brain. And so the atlases they put out are really very, very useful, and a lot of people are using them. But then in the second 10 years, they've gone off. Christoph Koch was hired, one of a CNS person, and he's the chief scientist. And so he said, look, we got to do the same thing for brain activity. And so now what they have is what they call a brain observatory. So if you have a project that fits within the set of constraints that they have, and it's the visual cortex primarily, they can record from different parts of the visual cortex with arbitrary stimuli. And if you have an interesting experiment to do, you can get time on it, and you can go up there. You don't have to do the experiment yourself. They'll do it for you. But the idea is so that you participate and you can see the data coming in, you can adjust it and so forth. And then, that you can take that home, and it becomes, as long as you're going to make that a public property, right? So that's the other breakthrough, is that up until recently, data never got out of the lab.
Yeah, and I think, data is certainly key to making such a thing happen. You need to measure all the relevant variables and the sufficient resolution, and you need to make it sort of jointly available for sufficiently large of people. But data in itself doesn't produce anything more than data, so to say, and for the goal, I don't even know if it is a goal to produce a really highly detailed model of the brain as a whole, that sort of functions and can do many things, and that sort of incorporates most of this data or much of this data. Is there a strategy for that, on the modeling side, so to say?
Well, there are many, many uses for data, and one of them is to allow people to analyze it with many different approaches, and each approach is going to see something different in the data. So just getting it out there is already an advance. Now, but the deeper question you ask, which is so the European Human Brain Project was a different beast. Henry Markham, as you know, was the director, and he was the one who had the concept and sold it to the European Union, and €1 billion. And I think that the goal was, in fact, to build a very detailed model of the human brain. Down to the last ion channel, I think. And I think that. That's an admirable goal in the sense that you'd learn a lot along the way, which is what I actually told a Nature reporter. But I think that it may be that at the end of the day, you actually have a perfect model, and you turn it on, and then it talks to you. What have you learned?
Well, I mean,
brains can talk, so
but this is where data comes in again. I mean, and this is the sort of the strategy for the whole body level that we sort of set out, that you sort of have this subdivision of labor for different aspects of the body, and each of these components that you put together needs to have been able to not only replicate data, but predict data a number of times and in fundamentally new directions for it to sort of be useful description of a component. And then you want the same type of predictive ability on the whole.
Right. So I have two comments, and here's I think the more important comment. No, I'm not sure which is more important, but in any case. Feynman once said that you don't fully understand something unless you build it and it works the same way that you thought it would. And so I think the ultimate test of whether our brain models are any good is if ultimately if we are able to reproduce and not just the function but the dysfunction when it goes wrong.
Yep.
And it will reproduce the behavior and the failure modes. That's what I think will have progress, and I think that's where AI is heading right now. It's heading in the neural direction, which is good, because we can help them. Now, but the other comment, which is something that Francis Crick once told me. He was a colleague here for many years, and we used to have tea every day at 3:30 when he would come to it, and we had these wonderful scientific discussions. And Francis had this mind that he just loved talking about science. I mean, it was a golden period in my lab, the 90s. It was just the golden period. ICA, dopamine neurons, Clay Reed and Peter Dayan. We had a wonderful time. But so I remember at tea one day, he said, Terry, you build these models, but you're taking them too seriously. They're not an end in themselves. And I said, well, Francis, what do you mean? He said that the real purpose of a model is to design a killer experiment that you would have never thought of doing that is going to give the game away. Right? In other words, it's a way of creating insights into something that then can be tested. And it's really the experimental test that will lead us to a better understanding.
Yeah, and I mean, quite often, not always, but quite often, once you know which experiment to do and you have done it, and you have seen it through the eyes of the model, so you know sort of the interpretation that the model supports, then you are like, of course it's like this. This is obvious. But before that, you didn't know which experiment to do, and even when you did the experiment, maybe you didn't know how to interpret. Maybe you had another interpretation. So I think this is sort of the ultimate goal, which is basically what Einstein said as well. That I mean, if you can't explain what you're doing and what you come up with to your grandmother, you haven't really understood it. So I mean, that's the ultimate. The complexity is only a sort of sub-step. Once you have found the principles and really proven them, then you need to simplify them.
Right, and those principles are very hard won, and you can count them on one hand from what we've known so far, and it's very different business from physics, where the principles from the principles you can derive everything. Right? Biology is completely different, for the reason I gave earlier, which is diversity. Every electron is exactly the same. It's like, you can do the experiment anywhere and you'll have the same electron in terms of the result you're going to get. And there's no two animals that are the same. There's no two neurons that are the same. So we need a very different theoretical conceptual framework to deal with that. And we're getting there. And I think these high-dimensional space, mathematics on high-dimensional spaces, I think will be very important for.
If I may comment, I agree completely about it. I actually, I have a degree in physics, but exactly I went into biology and other thing because physics were too bored. Everything was the same. But I think the one thing which is also I agree, I think with Georgi Bushakiu, who was talking about it, that the approach which we actually now follow is too much after the physics and engineering. And even what Gunnar was mentioning, it means that we are talking about inputs, outputs, boxes in between, and like Georgie was talking about it at some point, that we actually, our brain is the complex system, which actually it would be probably too simplified to actually say that there are some subunits which are connected by inputs and outputs, and we can follow and analyze them separately. It's exactly as you said, you have to have a very large data, and which is all everything, and basically it should be the whole body, because we should treat it as a very densely connected complex system, which actually it's difficult if you are thinking about network even or something like this. It would be very difficult to say where is the input to this part of the network and where is output from this part of the network.
So by the way, I love Yuri Bujaki's book. I taught it in my graduate class, and I completely agree with him. Almost 100%, not entirely, but what the punchline. Then this gets to your point about inputs and outputs.
Yeah.
The thing that really was for so long was missing. We turn monkeys into reflexes by giving them tasks where they have a stimulus and they have an output. Now, that allows you to analyze and correlate activity with different parts of the task. The trouble is that under normal conditions, animals generate behavior on their own. They don't wait for a stimulus to come in. The brain is active even when there's no stimulus. When you're lying in your bed in the dark, and your thoughts are going through your head, that is the essence of the brain. And until we have the tools for being able to get to the generators and what they're how that information is flowing through the brain and what is generating it, where's it coming from? That's really where we're going to make progress. And
but I would say that even if we are criticizing physics, but from the point of view of complex theory and the things which actually in the system dynamics when physics are actually working on, it's exactly not like this. So it means we are not in Newton or even Galileo type of the physics. We are talking also about the physics which are actually talking about emerging properties in complex systems. And even if we may model actually simple materials and other things, assuming that they are same, but we have a very big difficulty, and this is a problem actually in industry and everywhere, when you are trying to create more complex materials, which are actually with the kind of very small additions of different things. All electronics works on this and the other things. And if we are going deeper and deeper into it, we are starting to see that exactly when we input a more of diversity into the systems, we have to use different tools. And these are basically the tools from complex theory and this kind of the tools. And this is applicable, I think, to our thinking about the brain and even the whole body, instead of exactly thinking about this kind of the simple things that we have the input, we have a stimulus, okay? And there is the response of the stimulus, and based on the old physics, I would say, we are saying, okay, we want to repeat it. As you said, we are doing the same with this monkey, okay? We train them to do the same. And even, the whole thing when I was working in epilepsy research in clinical application, the point was that actually epilepsy is the nice thing for people who are talking about individual differences, because the point is that you cannot actually do a group analysis. And the whole point of difficulty of doing it in animals is you have clones, okay? You have animals, mice, which are clones. They are all almost the same. And in this case, that was early designed like this, because we wanted to actually repeat this, I would say, elementary physics, that we want to have the system which we know that it should behave exactly the same. But when you go into real, not only human, but normally animal behavior, it's not like this. So you should actually look into it differently. You might look, and that's why I like Bushaki approach, because you are looking about it that you have your own sort of patterns and other things, and we can call it memory patterns, we can call it other things, which are already in our brain. And then they are actually combined with the environment and what is happening in the environment, and then we have the reaction. So this is not a simple kind of input-output reaction. That's exactly the difficulty, even to what Gunnar was suggesting, dividing, for example, brain into the parts. I would say that was the old way of looking into brain, like, separately on visual cortex, auditory cortex, and other things. We know that there are diseases where people are actually seeing colors when they hear sounds. Okay, so in this case, you have basically the connection between these two systems, which probably are always connected, but most of the people during development actually separated them somehow. So I would say this needs a different type of modeling, not this kind of engineering approach, input, what happened in the box to create this output.
But you still do have that type of input output. I mean, you can consider individual cells, and you can sort of stimulate a cell and see what it's doing. I mean, a cell is in that sense
that's right.
System.
But in the brain and in the body, you don't have the cells isolated.
So
even if the isolated cell will do it, if you do it in the network, that would be different output. If you have the same cell actually connected with other cells. Sure.
Yeah, but that's part of the input.
What other cells are doing.
No, it's more complicated than that, because if you take two cells that come from the same part of the brain, tissue culture under the same conditions, you stimulate them the same way, you don't get the same response.
No
cells.
No, sure.
It's like and it's because each cell is self-regulating inside, and it's been responding to inputs, if hormones differently, and that means that it's on a different trajectory in this very high-dimensional space of what the cell can do or be. It's like a cloud of properties that they have, not just the same. And for epilepsy, I've actually worked with several people analyzing epilepsy data, Sid Cash at MGH in particular, on humans, ECOG. And there are many, many different types of epilepsy, and there's no two patients that have the same exactly kind. And it's true of mental disorders. Schizophrenia is not a single disease, mental disorder. It's a spectrum of diseases, and and there's overlap with bipolar, and there's overlap with depression and autism, and so it's very, very it's difficult to put joints on this system that we call the brain. It's very has so many different possible states.
But I would say, even on this level of the cell, I agree completely that if you might actually have the situation when you will artificially create the environment for the cell, that it will repeat itself. Even we are not talking that not every cell is the same. I am rather talking about it that actually, if you connect the cells into network, and the very large network, especially like the brain, when there are all of these interactions, not only electrical, but also chemical and everything, in the sense, it no longer behaves like this individual cell, and even if it behaves already as a part of the large complex system. And this large complex system doesn't respond even to the same stimuli in the same way. Because the system is actually never in the same resting state. Even what we are calling a resting state is never the same. So even if you put the same stimulus to the same kind of the complex system, it will respond differently because it was actually starting from different resting state. And we have this problem with brain, obviously, and a lot of biological systems.
So I can give an example of that. So I'm collaborating with John Reynolds here, who works on marmosets, visual cortex, and we have a paper that will be coming out soon in Nature. It's been known for decades and decades, and Bujaki is one of the world's leading experts on brain oscillations. Mm-hmm. So these are coherent patterns of activity where cells are firing, and if you wonder, why, and it's very controversial in the field about whether they are important at all for brain function or computation. A lot of ideas, but very hard to prove anything. So we did an experiment, and this came out of work that I had been doing with a postdoc, Lyle Muller, analyzing populations of recordings from ECOG from humans. Mm-hmm. We had noticed that during sleep spindles that there were circular traveling waves. The 10 to 14 hertz sleep spindles lasted for a few seconds that were repeating over and over again. And this is we know that the sleep spindles are important for consolidating memory, but how do you make progress? And so it turns out that in the awake animal you have waves too. You have waves traveling through the cortex, and you see these in arrays with the Utah array, 100 electrodes. It just covers one area called MT. And so what we did is we had an experiment where the monkey had to look at a weak stimulus where it was getting it right half the time, right? So it was right at the level of perception, of being able to react within a brief period of time. And then we looked at the phase of the traveling wave, and it turns out you could predict with very high accuracy from the phase of the traveling wave whether or not the monkey was going to press the button to report that it was able to see the stimulus. And so what this means is that these traveling waves have behavioral consequences for how the neurons are responding to the stimulus. So it turns out every single oscillation that has been looked at, for example, gamma oscillation 30 to 80 hertz or the theta oscillation in the hippocampus, they're all traveling waves. And in the hippocampus, the theta goes from dorsal to ventral, 4 to 8 hertz. So that immediately tells us that this whole idea of thinking about the neurons as being independent of each other, stochastically independent, isn't making any sense because of the fact that they're spatially organized. And that is a very different picture of how the brain organizes activity than the ones that we have right now. And we don't even know how to think about this.
Yeah. Well, I think that there are two sides to this. I mean, one is the fundamental assumption that if you would be able to exactly control the cell, individual cell, to have the exact same inputs as it has in the network, whether it would behave the same, I would guess that everybody agrees that in principle that's okay, even though it's a very hard experiment to do. And I think that just because it is like that doesn't mean that the best course of action is to fully understand each cell type before you start to aggregate things to the big picture. This is sort of the bottom-up approach, which I think has many limitations, even though the bottom layer should of course be there in data analysis and so on. You need to have the middle out approach where you sort of construct the behavior on a somehow resolution, and then you can fill in the details and you can sort of look at the top behavior. And I think that if you do that, then it might very well be so that individual cells are not that important to understand. You just need to understand sort of how a cell works, and then you study the networks, because it's in the networks that the behaviors that you're interested in happen. And I think that this thing that you talked about of stochasticity and variability, that you if you do the same experiment twice, or you study this another cell which is just next to it, it will behave differently. This is where it's so important to design time series experiments, because time series experiments allows you to have an n=1 paradigm, where you basically either for an individual patient or for an individual cell or for an individual component of whatever it is, if you have a time series for it, you can develop a model based on just the beginning of the time series and then you can have a model that is specific for that little system, and then you can use the rest of the time series for validating that model. And this is possible even though a neighboring subsystem or another cell will be, or another patient will be different. So you don't have to sort of do averages over many cells, but you can actually do individual models for each example, each copy that you have of the system that you're studying.
Yeah, no, the averaging is exactly the wrong thing.
Yeah.
You're absolutely right. You have to look at the individual cell over a long time.
Yes.
And you also have to be very clever. So here's something that is giving us a clue. Every time we've looked with finer and finer methods at individual cells, what we've found is that nature is not just averaging. The details really are important. And we stumbled on something a couple of years ago. So synaptic plasticity, we know, is very important for learning and memory. And we have various mechanisms. There's Hebbian plasticity, spike time dependent plasticity, homeostatic plasticity. And what nobody could figure out is what's the accuracy, what is the precision of plasticity? And in part it's because, you can't do the same experiment on the same synapse over and over again because it's changing. Yep. So you need to have some way of being able to look at a controlled experiment. And so we stumbled on it, which was a reconstruction of a hippocampal CA1 neuropil. And this is with Kristen Harris. And she's the master at EM reconstruction. So we did a complete reconstruction, very dense, of all the neurons, actually dendritic synapses and dendrites and astrocytes in this one little, 5 by 6 by 6 cubic micron piece of cortex. We noticed that occasionally when an axon went by a dendrite, it would form 2 synapses on the same dendrite. Same axon, same dendrite. Now, all of the models and all of the experimental data tells us that the change in synaptic strength depends on 2 variables. One, the pattern of spikes on the input, and the membrane potential on the outside. So some function, not very complex nonlinear function of the time history. Okay, so here's the ideal control experiment. Same input, same spikes coming in, same, because they were within a few microns, so the same membrane potential, right? Now, it turns out that the size of the synapse has been shown to be very accurately proportional to the strength of the synapse. So all we have to do is reconstruct the postsynaptic spine, it turns out, the volume. And lo and behold, if you look at the pairs and plot them against each other, they fall on a straight line with very small error. And a CV of 0.016 or something, which is telling us that whatever the synaptic plasticity mechanism is, it's incredibly accurate and precise, despite the fact that we know that there's a lot of probabilistic release and there's molecular variability and so forth. Yeah. But so that tells us that it must be important.
Yeah,
that such lengths in order to get such accuracy.
Yeah, and I think that this is an example of a thing that appeared in the chat some 15 minutes ago or something like this. That even though noise appears on a smaller level, there are lots and lots of control systems in biology, which means that you don't have to take all of that individual component noise and just add it together. But because of the control systems, certain features like glucose levels in the blood or temperature, and I mean there are many features that are highly controlled, which means that there are very noisy components in the control system, but you don't really have to consider them too much, because the control system will make sure that you have this very nice input-output or very nice homeostasis or something like this.
Yeah, no, there's no doubt that you have to have those in order to be able to remain stable. By the way, I once had a postdoc who did a really interesting project, which is, it turns out hormones do not just go up and down slowly. I mean, there are some, but it turns out that a lot of them are pulsatile.
Yeah, for sure.
And it's not at a regular frequency. It's not like, a sine wave or anything. It's irregular, and it changes frequency of day and night, and so forth. And it we did some mathematical analysis to try to understand, if we could figure out what was the purpose of that. But it occurred to me that there's that a system needs variability, right? In other words, variability may be a part of the actual design feature. Yeah. Maybe it's for calibration or something. You go up and down, and then you see how the cell responds to the pulse.
No, actually, as I mentioned before, you can actually think about some of the noise which we are seeing as the result of, it's basically the chaotic state of some very deterministic, maybe even system. The point is that actually, if we are thinking about how think about the system in the phase space, once again going back to dynamical systems, how make the system to actually respond very well to small or very range of the stimuli. You actually have to have this kind of the underlying seems to be chaotic noise because otherwise, if you are thinking about nonlinear systems who are in stable cycles, they are actually difficult to throw out from this cycle. You need to have a system which is almost on the border of instability. And this one is actually looking like noise. And to create such system, you usually have to underlying components, as you said, which on the level, when you are looking into each single component of the system, it will be very noisy. But then when you are looking into interacting components of the system, they will settle to certain states, but once again, they will be relatively easy to get to another state on the small stimulus. So actually, nature seems to actually make it right in the sense that exactly this kind of the system which are designed like animals to respond to something, they have this underlying noise and this unstable systems which are able to.
I wouldn't call it noise, but there are two really nice examples.
Yeah, I wouldn't call it.
One of them is the heart. So the heart, if you look at beat by beat intervals, it's irregular.
Yeah, that's right.
Unless you have heart disease. It turns out that when it becomes regular, your heart's going to fail.
Yeah, the same in epilepsy. Obviously, the pathology is when you are actually get over-synchronized and very regular kind of the brain activity. That's very pathological.
And brain. I mentioned brain oscillations before. It's unfortunate that is used. It gives you the impression that, first of all, it's synchronous, which it's not across the neurons, and number two, that the intervals are the same. It turns out it's very broadband, like gamma is 30 to 80.
Yeah.
And what it means is that, from cycle to cycle, the interval is changing.
Yeah.
And that that's I think the rule. I mean, very rare do you get something that is a perfect oscillator.
It's usually pathological if it is.
Yeah. I think this type of noise is sometimes also useful. I mean, there are many systems, both in biology and in engineering, which are designed to be unstable.
Right.
So the bacteria, for instance, that are so small that they can't sense the gradient can anyway, because their body is so small that they cannot sense the gradient because it's too much noise. They move in a random way. In all directions because they can't. That's the only way they can sense the gradient, by sort of moving randomly.
Yeah.
You construct airplanes out of stable regimes because that makes them faster to navigate. It means that they are more agile, so to say, and so on. So I think this whole thing of understanding stability, this is somehow what life is not. Life is somehow, by definition, energy flowing through a system to keep it out of stability. And it's in this place of out of stability that you somehow have the thing going on.
Yeah.
And,
By the way, there's another concept of robustness, which is a very important one. It is possible to use control theory to go outside of the range that a human can control. So fighter jets are not being flown by the pilot, except to give it commands, because it's in an unstable part of the control model of all of the problems that are going to occur at very, very fast timescales. So to get the best efficiency, you push yourself into areas that are not robust, where things can break down. And that's probably what's going on with epilepsy, with a lot of mental disorders, where things that had been stabilized went too far, too close, and over the edge.
Okay, so I think we've gone over the edge of time here, and to wrap this up, thanking everyone who spoke and for the late discussion, which was very fascinating, and we had a lot of people actually hung on till the end here. And we'll talk again soon, I hope.
Okay, thanks, Bill.
Thanks.
Sam, you did a great job. This has been a great workshop. I really enjoyed it.
Yeah, really great.
thank you very much.
Nice to seeing you, Bill.
Just seeing everyone.
Super interesting.